I will start. Maybe the talk is a little different from lecture 12. About pale green mutant of Chlamydomonas. And so yesterday I talked about the greening experiment, greening of Chlamydomonas, Y1 mutant, and transiently chlorophyll intermediates, geranyl chlorophyll, dihydro geranyl tetrahydro geranyl chlorophylls. So geranyl oxidoreductase or geranyl reductase convert the geranyl chlorophyll to chlorophyll. Okay, and this is a story about the mutant that deficient in the activity of geranyl reductase. Okay, and this work was done. Shirisha Koduru, she's from here, and her husband, Srida Nelpari, they together were some years ago in Okayama in my group, and then they did this experiment. And so it's published recently, granulated chlorophyll protein complexes in LHL3 mutant of the green alga Chlamydomonas reinhardtii. This is actually a collaboration with American group, Chris Nyongi. His also origin is from India, I think. He's a very excellent scientist in this field. And when we are searching for interesting mutant that has deficient in chlorophyll protein complex synthesis or chlorophyll synthesis to understand the assembly process of chlorophyll protein complexes, I got this mutant from the Chris Nyongi, a pale green mutant. So this is a phenotype. The cells are grown on agar plate, okay, spotted. And the LHL3 mutant, in the dark, it grows a little bit less than Y type, maybe a little bit less chlorophylls. So chlorophyll, that is pale, we call pale green mutant, accumulate less chlorophylls. So its color is pale green compared to Y type. And medium light and highlight condition, this is tap in the presence of acetate, so it's grow heterotrophically or under the light, photoheterotrophically. And the mutant GSM plate, that's the photoautotrophic condition. So we don't test in the dark, but it's middle light and the highlight condition. A Y type grow like this. In a higher light condition, it grow faster than middle light. But LHL3 mutant didn't grow photosynthetically. And this is the chlorophyll content per cells. Okay, the chlorophyll, microgram chlorophyll per 1 million cells. So Y type has 2 microgram. Sorry. Oh, sorry. Hello, this is Yujo Takahashi. I just called you. So this peregrine mutant didn't grow photosynthetically. And before we were working on some, as I talked to you the other day, there are some mutants that lack deficient in light harvesting complex. That is, they are also pale green mutants, but grow photosynthetically, although they need more light because that's deficient in light harvesting complex. But here, the similar type of pale green, but this mutant didn't grow. Something different. So the content of chlorophyll is just half of white. 8%. Then it could be some. Okay, I talk later, a little later. Okay, I will come back to this. This is actually. Okay. Okay. Then. Okay. Then we analyzed chlorophyll content. So we expect sometimes the peregrine mutant is chlorophyll B deficient or something like that. So we analyzed the pigment by HPLC. We extracted the pigment. This is Y type. So you can see chlorophyll B here, chlorophyll A here, beta carotene, and some other xanthils here. Not much. This LH3 mutant lacked chlorophyll A and B. Completely lacked. Well, that's strange because it's green, pale green, but it's green. Instead, we detected geranil chlorophyll A and geranil chlorophyll B. So yesterday I talked about this. So, and then if we add the phyto, so that is a reduced geranil pyrophosphate, it's very reduced. This becomes phyto pyrophosphate. So we add it. So the growth of LHL3 recovered partially. Okay. Photosensitivity is suppressed, and also even highlight, its glow grew a little bit more obviously. And for the photoautotropic condition, the mutant didn't grow, but if you add the phyto, you can see a slight growth, photoautotropic growth, right? But this is an anoxic condition, but the oxygen does not, removing oxygen did not recover photosynthetic growth. Then we analyze the pigment cells from the cells grown in the presence of phyto. The Y type, you don't see any effect of addition of phyto in the culture on the pigment composition. But HLL3, if you add the phyto, of course, chlorophyll B, geranil chlorophyll A and B is still present, but it's a little reduced. And chlorophyll B and A were separated. Okay, so this mutant is deficient in the conversion of geranil chlorophyll A and B to chlorophyll A and B. So granule reductase activity is deficient in this mutant. So we did the Western blotting using antibody granule reductase. So wild type, we could detect signal not in LHL3 mutant. So this is obviously activity must not be present because the enzyme did not accumulate in this mutant. And in the published paper, GGR, granule reductase, associate with LHL3 protein. That is comes from light harvesting like protein 3. So it has a transmembrane helix, which part is conserved, the amino acid sequence is conserved in this hydrophobic region of light harvesting protein. So that is named light harvesting like protein. These proteins also was missing this mutant. Okay. So we don't know which is which gene is the mutated. Okay. But anyway, the phenotype is obviously this mutant accumulate only granule chlorophyll. No dihydroxy granule, tetrahydroxy granule. Okay, this is larger. I make easier. Okay, you can see better. So we sequenced the messenger RNA of this mutant, and in the beginning, this part was done by Chris Nyog's group in, but they couldn't find any mutation in granule reductase gene. That was strange, right? The protein was missing, but activity is missing, but DNA is wild type. Okay, and then as I showed, L8L3, my collaborator suggested, then L8L3 genes must have a mutation. Then we tested the messenger RNA of genes of L8L3. So exon, 6 exons. So the first exon, there we found the deletion of 16 nucleotide in exon 1. So not GGR gene, but L8L3 gene has mutation. Okay. So if we did a PCR between primer between here and here, wild type has 136. Amplify the DNA product, but in the mutant, 16 nucleotide smaller fragment were detected, amplified, right? So also we, and then we complemented this LHL3 genes by nuclear transformation to see whether the phenotype will be recovered or not. Then if you have a recovery of the phenotype was complemented, so then we can confirm that the mutation, the gene with mutation is this gene. So this is, we need to confirm, okay? Not only showing this deletion, there may be, there are many genes that may be at the same time has a mutation, which gives as this phenotype. Okay, so we can specifically assign that this is a real mutation that gives us this phenotype. So we complemented, and we had many clones. We by complementing, we can screen the complemented strain and the photoautotrophic condition, because if the cell were not complemented, cell will not grow under photoautotrophic condition. So it's relatively easy, and then pick up the colonies that grew under a photoautotrophic condition, and then tested the accumulation of the protein of granular reactors and LHL3. So as you can see, DDR proteins are recovered. This is the wild type, the mutant, and this is the complemented strain. And also LHL3 give us two bands, because this protein somehow easily, one part is cleaved by proteinase, and then we detect always two bands. This is the whole protein, and this is some truncated protein. So the nuclear transformation of Chlamydomonas inserted DNA is randomly integrated into the nuclear genome. So depending on the insertion site of the insertion, expression level changes. So of course, as you can see here, some are very highly expressed, and then some are lower. So we have to select the complemented strain that expressed as much as wild type level. Okay. Then we analyze the pigments by HPLC, the wild type, chlorophyll B and A and beta carotene, LHL3 mutant, granular chlorophyll B. This is some mistake, right? Double C. I took this from paper, so sometimes happens. And the complemented strain totally lacks granular chlorophylls and only accumulate chlorophyll A and B. Okay. So we are sure LHL3 mutant has mutation in LHL3 gene.
The gradient is isopatric or binary for HPLC?
This is illusion?
Yeah. Like
this is a gradient. Yeah, we use a gradient.
So it's a binary gradient, or like isopatric, single or two?
One gradient. One gradient is from I don't remember the condition, but it changed the concentration of some more hydrophilic solvent and hydrophilic. The ratio is changed linearly. So this part is stamped field, so we didn't care about this stamped field. So this condition is not very good for separating stamped field. Only focused on chlorophyll. Then, because I talked yesterday the greening experiment. In the beginning of greening, transiently granular chlorophyll A, B, dihydrogranular chlorophyll A, B, tetrahydro granular chlorophyll A, B accumulated. Okay. How this granulated chlorophylls works? It's like behaves function like a white type chlorophylls or not. So because the LHL3 mutant didn't grow photosynthetically, probably photosystem 1 and or photosystem 2 do not are not functional. So to see this, we analyze the activity of photosystems 1 and 2. The first one is fluorescence kinetics measured with dual palm. Maybe you experienced the laboratory course doing this time the measurement of chlorophyll using a dual palm. So in the craminomonas, if you this is without measuring light, and this is measuring light on. So this is FO level, and actinic light is on. Then fluorescence increases and goes back down, and then becomes a relatively steady state. And then illumination, actinic light is off, then it goes down, and measuring light is off, then it reaches to the 0 position. This is Y type. And LHL3 mutant didn't show any this actinic light induced transient change of fluorescence yield, which means that PS2 activity is deficient in LHL3 mutant. Okay. And if we added a phyto in the growth media, slightly the mutant cell grew photoautotrophically. So if we tested the PS2 activity, so it's a blue line, you see a little activity of PS2. No. This is total less, but in adding a phyto, smaller, but substantial level of activity recovered. Because under this condition, some a part of the chlorophylls, small amount of chlorophylls accumulate, and granular chlorophylls are decreased, and instead the chlorophylls increased. So that's why activity recovered. Okay, and another measurement of photosystem 2 activity is oxygen evolution using oxygen electrode, and in the presence of the DCBQ, dichlorobenzoquinone. This is artificial electron acceptor. So we can detect specifically on the PS2 activity in under illumination. So wild type, this is oxygen concentration. So this is baseline. And if you on the actinic light, white type cell evolve oxygen, and if you turn off the light, actinic light, it stops. Okay. And the mutant, as you expected from the fluorescence induction transient kinetics, no PS2 oxygen evolving activity was observed here. So no activity of PS2. And if you add the phyto to the culture, partially activity was recovered. So this measurement is more quantitative, right? So almost half of the activity of Y type was observed in the mutant with phyto. Okay. Then how about PS1? PS1 also had active chlorophylls. So we used geolyotype spectrometry that we can measure PS1 activity using a cell. Okay. So this is a 705 nanometer absorption change. This is caused by photo-oxidation of pigment 700, P700. That is the reaction center of photosystem 1. So if you illuminate, AAL is actinic light. So this is dark. Okay. So no activity, but in the actinic light, P700 is oxidized, photo-oxidized. And then if you stop the actinic light, P700 reduced to the ground state. Okay, so this level is the amount of photosystem 1 activity. LHL3 mutant. This time gave us show a little bit about I don't know 20% of activity of Y type. So although we couldn't detect any activity of photosystem 2, we could detect small amount of PS1 activity. And if we add the phyto, the recovery is rather high. It's more than half, 50%, maybe 70% activity recovered. But anyway, phyto enhanced the activity of PS1 and the PS2. So eventually, photosynthetic growth of the cell in the presence of phyto, the mutant cell grow photoautotrophically at a reduced rate. That's obvious. Okay, so if you think about the greening experiment, in the beginning of greening, no chlorophyll and chlorophyll B accumulated, but in the geranyl chlorophyll A and B accumulated. So in the beginning, it becomes a little bit green, but from this data, the beginning of the greening cell will not should not contain show photosynthetic activity in the beginning. Okay. So this mutant is green, so pale green. So we expect chlorophyll protein complex is assembled. But we don't know detail of PS1 and the PS2 really accumulate because PS2 activity was deficient. Okay, so it may suggest that because the PS2 protein didn't accumulate or accumulate but non functional. The PS1 activity was there, but it's reduced. So that means PS2 is assembled in a reduced level or assembled normally, but the activity was low. So to answer these questions, we analyzed the protein accumulation by immunoblotting. There's so many data, so it's a bit confusing, but we checked the dark grown cells and medium light grown cells. Okay, the Y type, this is dilution, 100%, 50, 25, to see the signal intensity quantitatively. And minus means no phyto, plus is phyto. So this is LHL3 mutant, minus and plus phyto. And this is light, also with and without phyto. Then we compare the level of proteins by blotting. So PS2 is if you grow the cells in the dark, PS2, this is a mutant, okay? Here. PS2 is reduced about PSBA and PSBC, PS2 protein, and about 25% of Y-type level. based on chlorophyll. If you add the phyto, accumulation of PS2 becomes higher. And the concerning PS1, without phyto, PS1 accumulate more than 50%. Okay, and if we add phyto, the accumulation level recover to almost wild type level. So the presence and absence of phyto affect the accumulation level of PS1 and the PS2. The interesting thing is the PS2 accumulate without phyto, but we couldn't detect any activity. No, protein level was 25%, but activity level was 0%. So somehow with granular chlorophyll, PS2 was assembled but accumulated at lower level and no activity. This is the conclusion. And PS1 accumulate more than PS2, but activity is no, protein level is about 50% or more than 50%, but activity level was about 20%. So less than half. Based on PS1 content, PS1 activity was there, but about half or less than half. Okay. And also, this is a light harvesting complex, LHC1. And yeah, this is LHC1. And this is LHC2. This is LHC2. Because we have so many antibodies, so very often we test all the antibody we have. So that's why there are so many data, but they're mostly consistent. So LHC2, if you look at here, very similar level. So LHC2 is stably assembled with granular chlorophyll A and B. This is not the same as the PS1 and the PS2. So probably the structure of a light harvesting complex 2 is more flexible, can accommodate easily granular chlorophylls. So the LHC1, how is the LHC1? Also mostly more stable, but in the dark, they're stable. Okay, but in the light, some LHC1 are stable and some LHC1 are unstable. For example, LHCA3 here, without phyto, in the light, it's totally absent. Even dark, it's reduced. Others are almost similar. Somehow LHS6 is more than, but I'm not sure if this is reproducible or not, because we couldn't reproduce so many times using this so many antibodies. So the problem, if you work with so many antibodies, sometimes data becomes inconsistent. So you have to repeat more and more. So it's really becoming difficult to get very perfect data. But this is obvious. LHA3 is somehow unstable. Light. And PS1 is without phyto, the light completely delete accumulation. Okay. So this is the other our published paper. LHS3 becomes unstable in the absence of PS1 deficient mutant. So this data is consistent. So I'm very much sure that this absence is reproducible. Okay. So another thing is PS2 accumulate less in LHS3 mutant, but still in the light, PS2 is accumulate similar level, but PS1 is less reduced, but under the light, it's gone. So PS1 is very unstable in the light. This is interesting phenotype because usually in the white type, PS2 is photosensitive, very photosensitive, but it's resynthesized, it turnover, so steady state level of PS2 in under the light, not if it's not very strong, the PS2 level is maintained constant, although PS2 activity decreases, but protein level is constant. But here, PS1 is in the white type is stable. The no activity decrease is observed if the light is not extremely high, it's moderate light. But here, PS1 is more light sensitive than PS2. This is interesting, I think, phenotype. Okay, so mutant, from this many data, it's sometimes confusing you to see what is interesting and what is we should focus on which phenotype or not. Okay, and then this experiment is analyze the stability of protein. Incubation time of cell, 0 to 6 hours, wild type and mutant, and this is no addition. So protein level is almost constant, almost. But if you add the chloramphenicol to block the synthesis of chloroplast proteins, chloroplast encoded protein, and the cyclohexamide, which blocks the synthesis of nuclear encoded protein, to see the stability of the protein. And chloramphenicol, this is a PSBA is a chloroplast encoded protein. So the synthesis of this protein is blocked by with chloramphenicol. It's going down. Okay, relatively PSAD is stable. Cyclohexamide is nuclear encoded, but LHC8 and 3 are nuclear encoded protein are stable. So LHC is stable. So this means that PS1 protein, this also going down. So PS1 protein is unstable, although stable in the wild type, but the mutant unstable. PS2 also unstable, but probably resynthesized. So that's why it's balanced, equilibrated. This is a phenotype. And we also analyze the chlorophyll protein complexes. This bicycle-state gradient in the dark, a wild type, LHL3 mutant, and the phyto. So the difference was this mutant has reduced level of A3. This is PS1 LH1 supercomplex, as expected, and has more here. It's stronger, so compared to wild type. And the middle light, wide light, PS1 electron supercomplex disappears as expected. But level of LHC2 nearly constant. So the increase of this band in LU3 is due to this content of PS2 and also LHC1 dissociated from PS1 because PS1 is reduced. So this happened. Okay, so this is a phenotype of the GGR LHL3 mutant which lacks the GGR activity. I think this is the last slide, but I skipped this. We just purified with the HA tag, but I skipped this because it's no, it's yeah, almost right. So in my opinion, the granule chlorophyll is the phytol is flexible. The granule is additional 3 double bond. So it's like this rod. Its tail part is less flexible. So if you accommodate the chlorophylls in the chlorophyll protein complexes, sometimes this tail must, if it's flexible, you can easily accommodate in the chlorophyll protein complexes. And if the tail is somehow rigid structure, it's sometimes difficult to accommodate properly. So position may a little bit change. So that may affect the stability, or that may affect the activity. That's happened. So during the greening experiment, we observed the accumulation of granulate chlorophylls, but these are not really functional yet. So we have to wait the greening to continue to express fully the cell express full activity. So this is we can get this conclusion from this mutant experiment. Okay, I stop my talk and ask you a question. Yeah.
Is there any structural?
Microphone.
Is there any structural data this incorporated geranyl chlorophylls in photosystem 1 or photosystem 2?
Structure.
Yeah. Any structural data?
We have not. We want to do structural analysis, but so far we have not yet studied. But I'm planning to do so because we can separate these complexes easily, and PS1 is slightly active and PS2 is totally inactive. So from the structural analysis, we may have some information.
Yes.
Why? Well, probably reaction center chlorophylls, P680 or P700. We are curious to know the structure. And also acceptor side. Yeah.
And you're in functional, that kinetics activity, PS2 activity is completely absent.
Yeah.
And PS1 activity is bit there, slightly.
Yeah.
But Western blot data, PS1 proteins are almost
reduced, but its accumulation is higher than the activity.
Okay.
For example, 50% level of wild-type level accumulate, but activity was 20% of wild-type level.
And according to your view, it must be you showed the absorbance of these chlorophylls, like geranyl and all, in your previous class. The absorbance is same.
Yeah, exactly, because absorption is determined by the structure of a chlorine ring structure.
Yeah. So you think it is because the bonding or the energy transfer from this geranyl, or what could be the reason this activity is completely? Either it is the bonding efficiency or it is the transfer efficiency, because absorbance is similar.
Yes, absorption is completely similar because the tail does not affect the structure of the chlorine ring structure. And this tail is not involved in excitation energy transfer. So mostly this hydrophobic tail is fixed, is used to fix the chlorophylls in the chlorophyll protein complex. That is mostly chlorophylls are located in a very hydrophobic region. And its protein moiety is very much compact. So this tail is somehow fixed in this very condensed structure. So flexibility is depending on the position of the chlorophyll in the chlorophyll protein complex. So somehow some position this granule tail is very difficult to so position may change a little bit. So that affects the excitation energy transfer or charge separation. The chlorophyll is involved in not only primary donor and acceptor, accessory chlorophyll, and also in the case of PS1, I didn't talk, but it's phyroquinone is also double bond is removed by granule reactor. So that's this is also intermediate acceptor of PS2, not plasquinone. The fair fighting also affected probably.
Very
so
interesting work.
If you want to know which part was affected more significant, the most significant is if we can determine the structure by cryo-electron microscopy, that helps. We could know.
Nice.
Yeah, yes.
Thank you.
Any other questions? Okay. A little bit too detailed, so maybe it's not easy to follow sometimes. Yeah. Microphone.
You said there's no GGR activity in LHL3.
No.
Why is it? How are they both related? Because the mutation is in the LHL3 gene.
The question is that this mutant is LHL3, but why GGR is absent? So we can no. This I didn't tell you that HA tag using HA tag we purified, and then GGR was co-purified with LHL3. So LHL3 has transmembrane helix. Then GGR is soluble protein, but it is too associated. So probably LHL3 link GGR to cytochrome membrane. And without LHL3, probably GGR becomes soluble. Then somehow it becomes unstable, probably digested by proteinase. That's why the GGR does not accumulate in the LHL3 mutant. So this happens. Sometimes, of course, without LHL3, it could be, GGR could be accumulate, but this was not the case. And also, GGR must be very close, attached to cytochrome membrane, probably, because granule chlorophyll must be present near the cytochrome membrane. Okay, so no other questions? Okay. So maybe I'm going to Kerala until Tuesday, so I come back Wednesday. So if you have questions, then
Am I audible? Hello. Okay. So the topic which I am going to discuss little bit more on light arresting, degradation of light arresting complexes. Degradation of LHCs during acclimatization and senescence. Maybe a little bit in the inside will be little bit different, but mostly we'll be talking about light harvesting complexes. LHCs means light harvesting complexes. So this might have discussed with the HRO, but I also told you in the last first class, the light arresting complexes here. This is from plants, and this from the lower organism, cyanobacteria. So this is the cyanobacterial thylakoid schematic representation, and this is the schematic representation of higher plants and algae photosynthetic complexes. So since we are talking about light arresting complexes, how they arrange, just we'll discuss. So this membrane complex, thylakoid membrane, you see that the linear structure where comprises with the lipids. You can see the bilayer, lipid bilayer, this one. Then you have we also discussed last time, now we have a core complexes that's called reaction center, and they have a D1, D2, CP43, 47 like protein and their respective genes. And there will be, when the light is absorbed by the plant or algae or any photosynthetic organisms, that would be harvested by light arresting complexes. Light will be absorbed by light arresting complexes. So these are the first protecting a pigment protein complexes around the core complexes. So they will be the one it harvest and transfer that the light, to the photons to the reaction center where water get split, whatever the water, and you get oxygen and protons, and we discussed last class also the hydrogen energy will be coming through this photolysis of water. So if it is coming to the cyanobacteria, so they have three light arresting complexes, that is allophycocyanin, this is the round pigments, and phycocyanin, this one PC, and the reddish color, the one phycoerythrin. So all these pigments are present in cyanobacteria, and also they will be also present in red algae. It's anaxigenic photosynthetic bacteria. They also have similar kind of pigment, but they may not have allophycocyanin, but phycoerythrin and phycocyanin is there. So last class when we discussed why Niagara is bluish color, why most of the lakes are very colorful, why is called Red Sea. So Red Sea is, why it's called Red Sea? Any of you can tell? Huh? It will be having more blood. No? Huh? Huh? Algae. It's algae, but no, as I said, it can be anthocyanin and other thing, but mostly because of the phycoerythrin. So that's why it looks like a Red Sea. It comprises the bacteria, algal anaxigenic bacteria are present. When the light is it's like when shine onto the water, whatever it, because of reflection, you can see Red Sea, and Black Sea also there. Red Sea, Black Sea, whatever, most of them it will be bluish. That's why it's called cyanobacteria, blue green algae. Okay, so now these are the light arresting complexes. So now similarly in higher plants also, higher plants are algae. This is the core, similar to this This is the core reaction center, and you have a light arresting complexes. So light arresting complexes surrounded by these, the surround, to the core. Similarly, you have photosystem 1, and also having this same kind of core complexes. You have a light arresting complex. The number of light arresting complexes varies from plant to plant, and also algal have a more light arresting complex because they live in deep water or fresh or the sea water, where it require more light arresting complex. That's why they have more light arresting complexes. So if you look at this one, they say that these are the proteins and their respective genes. You represent in italic letter, then it will become a gene. Similarly, allophycocyanin, they may have several encoding genes. These are all, spicocyanin, several encoding genes. Picorithin, there are many, several encoding. But light arresting complexes, this is about 5 in photosystem 1 and about 6 to 7 in photosystem 2. These are the light arresting complex about the structure. So now little bit we'll go about physiology and other the biochemical part. So now if you see this video, plants exposed to various kind of environmental conditions. So it was mostly you see that then when seasonal changes, it changes turn into from spring to it will go to autumn, where you see a lot of changes on to the plant. Now they're green, now it will become yellowish color, we just discussed, like become anthocyanin and other carotenoid pigment they convert. And now this is a cycle. So I don't know, winter, and most of the and the question comes here, maybe you can answer me. Any of you been to Himalayas? Yeah. And winter season. In winter season, and where it gets snow of this much height, and most of the European and US and Canada, they will have a snow full of snow. The snow, and you don't see any leaf. There won't be any leaf. There won't be any except pine trees. All trees doesn't have any leaves. The question is here whether they can perform photosynthesis, how they live. They know photosynthesis. How they get food? Except pine, all other plants, apple, you say that apple, mostly an apple, berries, all other, any other crop. So they don't perform photosynthesis because there is no green stuff there. Everything is gone, all leaf fall. So it looks like this, you see the snow and no leaves. Of course, anyway, so the water will be there in all, they will be alive. Once the season is over, spring comes, then it blooms and you get the photosynthesis. Even if the sun is there, in places like US and Canada in the winter season, they will be sunny, but cold is about minus 10 degrees, so they cannot live. One thing here is, there is a it won't be hibernation. Hibernation means they don't do, but they have a cyclic electron transport where they don't need any light, but they do circulate energy through metabolic pathway. Metabolism will be active. Through metabolism, you get the ATP. ATP will be generated. It will be a cycle. The electrons come degraded, Kelvin-Benson cycle or something by Maillard biosynthesis, where the electrons will pump into it, makes a plant continuously ATP generated, and plants will be alive. So plants will be alive, but photosynthesis happens, but not active linear electron transport. Okay, so now, so during that time, what happened? Plants, as I said, this is called you can also call acclimation, or these all these process exposed, or in all these process will be can see in during the plants or algae exposed to different conditions. So here there is a called non-photochemical quenching. Non-photochemical quenching. So the meaning is, here you can see here. So let's take from here to here. That means seconds, minutes, hours, and days exposure of the plant or the algal system. There will be several things happen. The seconds, if you see the seconds, so now this is called short term, and long term is basically hours to days. So during that time, these are all the events can happen. Change in electron transport, formation of delta pH, that I will tell you a little bit later, and picky pull reduced state, and photoxis, and deepoxidation of xanthophyll cycle, and increased xanthophyll cycle. If you go into little bit more hours together, you can see up to short these will be there, and longer duration of the the plants, they will be adapted to several these mechanisms, transcriptional changes, protein degradation, and PS2 repair. That's what I am going to discuss. The light arresting complexes will be get degraded. That was a change in the antenna and change in stoichiometry of PS2, PS1, and accumulation of a lot of antioxidant. This can happen. So the non-photochemical quenching is basically, again here you can see here what is non-photochemical quenching. So if you look at the thylakoid membrane, so thylakoid membrane, as I said, the linear, where you have a lamina under the peripheral and the lumen. Lumen is downside. When the electron is pumping from top to down, the protons, whatever is the protons which you talk about, hydrogen ions, the hydrogen ions will be pumped more towards peripheral to the luminal side, and where that will become more acidic. The down will be more acidic. The pH will be acidic, and the acidic is the sense is a favors to the chemiosmosis. Chemiosmotic means again the production of ATP. So the chemiosmotics is a down state, down of the lumen, it goes down and goes to the ATP motor. So then where you may have many things happen, a lot of the cyclic electron transport will be more activated, but however the delta pH is nothing but a luminal pH become acidic. During that time, there will be a QE, that is one, QZ is another one, and QT is other one, and QI is another. Three types of the four types of adaptation. So QE is nothing but this is called delta pH will be more increased. Through that there will be a specific genes or proteins, that is delta pH, LHC SR, or is also called PSBS, photosystem reaction center protein, that will be overexpressed by acidic pH, and yet this component will be non-photochemical quenching. There is another one, non-photochemical quenching. Here is the one, this is the one. There is a pigment called carotenoids. I said we talk about carotenoids. The plants, when it goes to any kind of stress, the plants is trying to convert the chlorophyll pigments to the carotenoid pigments, where they will be photoprotecting. Photo means when the light is more, they try to get protected by photoprotecting, where the zeaxanthin is a pigment, convert more to excess light will be quenched. Quench means reduce the light. So that means even if it is falling lot of light on the quantum of light, it try to take only the light which is required, and where the zeaxanthin pigment is the one, it controls the light. So that is the meaning of that. There is another one, this is called state transitions. There is one that we will be discussing later. There is a enzyme called STT kinase. That kinase getting phosphorylated, that is called QT, is the one that we will discuss bit later. And this another one, if it is, let's say you can say that cat in the room, you hit the cat, just you irritate cat, cat won't do anything. If you irritate cat, what it does? It will become pulley, it will become tiger, right? Cat will become tiger. So same thing here. So what it does, you keep stressing the plant too much, it get damaged. So that's what it's called photo inhibition. Even photo inhibition also, it can be, there will be some repair mechanism is there. Still, there will be trying to protect. That's what, if somebody is hitting you, that means, when we hit cat, it try to protect he or she himself, so that, there is something still, I will be alive. So this is the way, it's called, the photo inhibition. So these are the, the mechanisms the algae and plants can protect themselves. So there is a other mechanism, as I said, redox regulated phosphorylation of light arresting complexes, which is the one called state transitions. So now you all know that by this time you might have learned what is PS2, what is photosystem 1, right? HRO has described. PS2 absorbs light at which wavelength? 680 nanometer. PS2 absorb light at 700 nanometer. Why so? Right? That also I discussed. But this difference, when you see the electron transport chain, the Z scheme, you call it as a Z scheme, there is a voltage, significant voltage difference in there. This voltage difference from photosystem 2 to photosystem 1 is balanced by this so-called state transition mechanism. That's called light, the redox regulatory mechanism. So it will be like a motor, the transport system, it's going like a circle, which we're going to discuss here. What it does here. So now the photosystem linear electron transport, if you get photo 680 nanometer, it excites the electrons and goes to the PQ pool, then PQ pool to the cytochrome B6F complex, then it goes to the photosystem 1, it will be like that cycle. So the PQ pool here, what it does, it will be, this is photosystem 2 light arresting complex, two of them. And let's take a photosystem 1. There is a photosystem 2 and photosystem 1, two different. So when the system required to balance the energy, so when it is now let's say if you're going outside, if you're going to the outside, plant exposed to fluctuating light conditions, there will be fluctuating light. That means rainy season, you get a hard, I mean yesterday, day before yesterday, we could have seen it will be shiny, then again it get reduced, or even if it is a gradually increased light. So what it does, the PQ pool get reduced, that means plastoquinone pool is oxidized reduced mechanism, that is redox regulated mechanism. It get oxidized and reduced, oxidized, reduced. When it getting reduced, what it does, through LS2 kinase, it get phosphorylated. You see ATP will become ADP, that means one of the phosphate group is the ADP, diphosphate, from triphosphate to diphosphate. The one of the phosphate group is going to attach here, that is this, it will become the light arresting kinase. What it does, the kinase of LHC2 and phosphate group is attached. This phosphate group is now is going to be here poised, that means they get together and it will become a photosystem 2 here. Photosystem 1, I mean poised means with the photosystem, with the LH2 is associated with always photosystem 2. When it get phosphorylated, this in general biochemistry, if anything, any phosphorylated form is stable or unstable? Any phosphorylated form is unstable. So unstable molecule cannot be, they'll be like, right, if you are tensed, right, you will be unstable, your mood will be fluctuating. Similarly, here also, when it is getting reduced form, reduced form never will be sit with the photosystem 2. It has to migrate to photosystem 1. That's what it does here. Photosystem 1 light arresting complexes, complex it form. So then what it does is called state true condition. That means when light is exposed by the plant, that means mostly blue light, that means visible light, so-red region, this takes the light and get phosphorylated, then migrated to the photosystem light arresting complex 2 will migrate to photosystem 1. Then then what it does, my job is done, right? The vehicle, let's take from here to Hyderabad to, let's take Rapido, right? You can send Rapido food delivery. So food delivery, deliver, and what it does, that guy will go empty. So empty, what it does, again you go to whatever Swiggy, whatever Zomato, you go to again shop Paradise Biryani, get Paradise Biryani delivered, again he has to go and deliver again. Similarly, what it does, it get oxidized, the picky pool get oxidized, and this using phosphatase enzyme. That is a kinase enzyme. Kinase enzyme is always ATP will be become ADP, phosphate group is attached. Now here phosphate enzyme, what it does, again ADP will become ATP. So the phosphate is removed and ATP will now be formed. Then all of them, these will be also dissociated. The light arresting complexes, they will be dissociated. Again, this is the cycle. This is called state 1 transition. Oxidized is state 1, reduced is state 2. So, and different light condition. It requires different light condition. Here is, it favors to photosystem 2, it favors to photosystem 1. Photosystem light 1 is longer wavelength and photosystem 2 is shorter wavelength. So this is called state transition mechanism. Through light arresting complex, it happens. I mean, little more to schematic, more representation. So this is the one, these are the kinases, STT7 in algae, STT7 is in plants, and this is reduced condition, and this is the oxidized condition. This is PPH1 and TAP38, these are the phosphate enzymes which is involved in oxidation. You can see that light arresting complex is attached. So then it does, the photosystem 1 is going to attach here, where you can see here the light arresting complex 2 and light arresting complex 1, this phosphorylated form is attached. Then when it get oxidized, it will go, only light arresting complex 2. So this is the mechanism of state transitions. Okay. So we also showed this one from, this was one of my collaborated data, and also I also showed this one, my 20 years back story. When it is, this is the cell, you can say this is the chlamydomonas cell, this higher plant, Arabidopsis, sorry. So this is a cryo-electron microscope. Anything structure you want to see, cryo-electron microscope, you can, using that one, what we showed, how it is a regular, this entire light, whether, it's experimentally you have to show whether it is true or not. So you can see this is the photosystem 1. The phosphorylated form of light arresting complex is migrated. That's what, this data shows. So, just want to show a little bit more representation. So this is a PQ pool. Get kinase is activated by kinase activates the LHC2 phosphorylation. So what it does now, so it get excited, it goes and bind to the photosystem 1. So this is called state transitions mechanism. Happens every plant, every photosynthetic organism. In phycobilisomes, if it is even in cyanobacteria, phycobilisomes will be there. Even in anaxin bacteria, this system is there. So then what it does here, when it is migrated to, this is the one, you say that for state 2, now it has to be migrated to state 1. So now what it does here, excited, and this goes and backs to dephosphorylation, it goes and binds the light arresting complex, go and migrate to the photosystem 2. So some of the data I would like to show here, but not much, from our data or from our laboratory, how light is induced state transitions influence the light arresting complexes, how different abiotic stress can induce state transition that we wanted to understand, and what kind of signals are playing major role to inducing the PQ4. So the simple technique which low temperature is done today, tomorrow, yesterday, Monday. Okay. You will be showing this technique, fluorometry, simple fluorometry, where we show the difference between state 1 and state 2 complexes. That means we induce physically, that means light conditions, and then see that when photosystem 2 light arresting complexes, when it get excited, that means it get phosphorylated. The phosphorylated complex has to migrate to photosystem 1. That means, so let's take the balance. So if I have like this, so if I have put more balance here, so my weight will be, what happens? My weight will be increased. The weight get increased. So then what happens in the experiment, simply in simple terms, we need to show that one. The photosystem will be, it will become less, but we have normalized here. And you see that photosystem 1, this photosystem 2, and this is the photosystem 1. When light arresting complex is more, that means height is more, or the weight is more. You can see here, from here to here, there is a microscope. The junk of, we'll see here. So this is called major light arresting complexes, where to want to show this one biochemically, what we are showing here. So the LSC3, LSCB1, and LSCB2. That means 1, 2, 3, 3 subunit, 3 genes are very important for trimeric complex. This trimeric complex, which light arresting complex is very important and which one is getting phosphorylated. That's what we wanted to show. So here, all of them are getting phosphorylated. B1, B2, B3, all of them are getting phosphorylated. We are matching here. So, but and when you see that this is the antibody we used, anti-phospho-3N. So then 2D electrophoresis, the technique is called one-dimensional electrophoresis and two-dimensional. It's called isoelectric focusing. We separate the proteins, then go with 2-dimensional on the SDS page. That means you can separate based on the molecular weight, where we showed here, again used antibody work and identified the LH. I don't want to go deep into the story. The LH2B is a major subunit or protein, is inducing state transitions. Of course, the other proteins are also inducing, but because of that, there is, you can see here, this is not state 1, 1, 2, 3. When you see here 1, 2, 3, 4, 1, 2, 3, 4, there is one of the isoforms getting complexes. That is an indication that LH2B play major role, really major role to induce the state transition. That's the meaning of that. Okay, then we separated complexes where it is getting associated with to show more biochemical data. Sucrose density gradient, the HRO, he has showed many things, and where we showed, the S2, they have an additional band that is called light arresting complex. Photosystem 2 to photosystem 1 is migrating. So let's take, if it is a dark condition, plants also exposed, let's say, talk about there is no light. what happens? That's what we wanted to prove. So can you understand what is it from this? From this? Huh? No, from these, I mean not the subject, I am not talking subject. From this, what do you expect? What do you can give a conclusion? No, simple. These people are walking straightly, right? And these are all visually challenged people. They are also walking. That means in the dark cycle, the dark, there is no light. These people also have to live the life. So they are also walking with the stick, with the support they can walk. They are better than us. They know really. They also follow discipline. But with this traffic, Indian traffic, so they could able to cross. I mean, I don't know whether it is Indians, but it looks like Indonesia. So they're able to cross the road. So they also can cross. I mean, there are bright people. They can see the light. They are also. But the meaning of the story here is, so even if it is stress, plants are they become a. I mean, in the regular condition, that's called state condition. In the dark or in the fluctuating conditions or highlight something stress is there, they also plant can survive because of some mechanism that is also called state condition. That is called dark induced state condition. So whether it is true or not, we showed, we used Arabidopsis. So here we use a temperature. Just increase the temperature can induce the state condition. They perfectly induce state condition. So in the mutant, they're not performing state condition. That means LHC2 kinase is still activated. The question, this is a lot of, to convince the reviewers, it was so difficult. These are all our laboratory data work. Convincing reviewers, this was a plant journal which we submitted. We are not able to convince them, but they asked us to do double mutant, but we could not be able to do it. It's Sridhar who was the first student who did all this work. Anyway, so but we got a lot of the convincing data. In the end, people have agreed that there is dark induced state condition, which we have discovered from our lab. So when any kind of stress also induce state conditions, but it may not be mimicking, but LHC2 is getting phosphorylated. That's what we wanted to show. This is the LHC2, they're nicely getting phosphorylated. But there are many other things happening. I don't want to give you more. I mean, this is the same thing. The phosphorylation is happening, and it's not happening in the mutant. Wild type it is happening, and mutant does not happening anything. And similarly, when you induce, you see that again stress, very nicely inducing, this individual LHC subunits of LHCs, they are getting phosphorylated, all of them. Okay, role of, I don't want to, this is a lot of biochemical data. We did a lot of confirmative analysis. We used various inhibitors to show whether it is true or not, how they are getting. This is where we showed the really signal, what kind of signal is coming, where it is coming, the DCMU and all. They can inhibit the phosphorylation to show many things. I'm not going to discuss about this one. So this one also it is not required. Then we showed the migration, whether it is guide. So you can nicely shown that there is LH2 is getting, you see that 40 degrees nicely coming out. This is the purified complexes from sucrose density gradient. And we showed several other, the parameters to understand. This is a cyclic electron transport. This kind of thing is cyclic electron transport. So what we have connected, there is a linear electron cyclic electron transport. In the wild type, this is a linear. When it goes to the 40 degrees, when you incubate, cells are, the leaves, when you incubate 40 degrees, they will induce state conditions, the state condition through cyclic electron. What we have showed from this data is, in regular light conditions, they don't induce state conditions. when it's a stress conditions or in a 40 degrees or anaerobic condition or you induce any other stress condition, they induce cyclic electron transport, and in turn cyclic electron transport gives and activate light arresting complexes. Then for the PQ pole, that is a plastoquinone pole, get reduced and light arresting complexes are activated, then the electrons will be, then through that the phosphorylated light arresting 2 can migrate to the photosystem 1. We used, I don't want to show, these are all the mutants, lot of mutants we use to show the migration. These are the two schematic representation. So what we said, through that there will be a cyclic electron transport in dark cycle, not light conditions. This is the cyclic, these routes are cyclic electron transport, PGR1 and PGR5 and NDH. The electrons are pumping through this one, and through this PQ pool is getting reduced. This one is the reduced, so that whatever you are getting, and we showed here linear teletransport and cyclic electron transport. So the photosystem 2 is getting reduced through the cyclic electron transport. That's what we wanted. I will skip here all this one. You don't need all this one. Yeah. So finally, what we showed, these are also a schema in algal system. So you will have a, we induce heat and show that the LHC2 getting phosphorylated and associated with photosystem 1. You can see here, this is what in an algal system also it is happening. So further, this will be a next Monday. I am going to discuss the other thing, the chloroplast adaptation or chloroplast development in light condition. There is one more topic that I will discuss. So if you have any queries or clarification, I will be happy to clarify your doubts. Yes. Yeah, that's very good question. So that's what it tells. Your spectrum, the solar spectrum, even an algae or plant, when you change different light conditions, the blue light is fine. Blue light is always good for the blue light. That's what when you light shows there is the WebGR color, but plant absorbs two lights, that is a blue and red light. These two are the major one. So blue light and red light combination, if you give it, it's good. If you give only blue light, so you are only favoring to photosystem 2. You are not favoring photosystem 1. So you have to give blue light, or even if you can give fluctuating, this is fine. They can grow nicely. So let's say if you give only red light, plants cannot grow. Only specific, let's say 700 nanometers, plants will be stressed. And if you have to give, that's why we give always solar, the 400 to 800 nanometers or 400 to 750 nanometers. Did I answer you? Yeah. Yeah, exactly. Yeah, you are right. This is called again adaptation. So the pigment and algal or plants, now you need to have let's say a commercial importance. You can give, you are changing, something you are changing, you are getting something out of it. So you want to have a, let's say now where Pavitra was she never observed any pigmentation. That means we are giving a right condition of environment. We want to have a, let's say a pigment, then we change the conditions, either light or nutrient. So light, you go with red light. What to give, if you give more of red light, mostly pigments will get changed. Chlorophylls will be changing, it has to be changed, chlorophyll, and it favors more of photosystem 1. So there will be, if it is more photosystem 1, you may have to have a lot of xanthophyll pigments will be accumulated. More xanthophyll pigments will be accumulated. Let's take if you give a blue light, more of blue light. So there will be a condition of lutein will be increased. Mostly lutein will be increased. Likewise, you can change for a commercial purpose. Yes, you have to change to get the pigments. You are not benefiting the algal system or plant system, but to human selfishness, you want to get something. Yes, you have to change something. The parameters you get different conditions. That is the same thing. Like there is Urban Kisan company. There is they do vegetables, ground roof vegetables, where they do all these changes. So what they say that the any amaranthus family or greeny leaves. So you get nice anthocyanin color, complete anthocyanin color. What I work for them as the consultant. So we change different light conditions, and they get all these like a beetroot color leaves. That is only because of we didn't manipulate any genetical manipulation. We manipulated light, got different color. So that is now in the market. You can see, have you seen any of you? The leaves, there are different color of leaves. That is only because of light. If you change something genes, yes, it can be easily. But naturally, you can pigment it. I mean, you can do it, you can increase the pigmentation. Of course, composition is also different, right? Two people, may have two different kind of, understanding. So like, let's take a chlamydomonas, okay? Hard called chlamydomonas, right? Or chlorella. And, you take a hematococcus. Hematococcus only accumulate astaxanthin, right? Because their expression, their genes, the pool of genes are more for the hematococcus. So the same gene is not in the chlamydia. So there was one, I think last class, Vijay, you presented, right? The chlamydomonas gene put into, the chlamydomonas, not chlamydomonas gene. Yeah, they put it in the chlamydomonas. They have expressed astaxanthin here. So, nanochloroplast genes. Okay, chlamydia to nanochloroplast. So, especially those who are algal system, they live in different environment conditions. They have to adapt to that environment condition. That's why they evolved different pigmentation. But ultimately, in the beginning, every favorable condition, they will be happy to grow. But if you give something kind of light or nutrition deficiency, now they will be obviously they will have a different genes will, even though genes are there, their expression level will be different. Let's say Ichiro Takashi has come, so he expressed differently. He was staying in Ella Hotel. He was not able to cross the road. We can cross, we can run, right? So we adapted to that environment. He could not cross, and he came back again hotel. I said it's not my cup of tea to walk on the street. Clear? Okay. Any other questions? Yes. No, this is a technique. Electron microscopy is a technique. So where when you go to Kelvin, low temperature, I mean they use a liquid nitrogen. So liquid nitrogen generally what it does, it's a freezing condition. When it is frozen the any kind of cells, not only the photosystem, you go with any cells isolated complexes, the reflection rate will be higher and the visibility will increase with the lasers. With the lasers, it will be like a shiny, more shiny. The visibility inside will be much better. So based on the section, you take a section and you freeze it and expose a laser, and you might see much better reflection. So whatever inside is there, that's what that nowadays That is the older, about 20 years back, 15 years back research, or 20 years back. So, but now even much better, you can look at atomic level inside. Atomic structure is they now determined by, Pavitra is doing the same research what with the German collaboration. We are looking at atomic level, what at subunit level. That is the advantage with the technique. Yeah. Is it clear for you? Yeah.
There are two types of state transition. One is light induced and one is dark induced. Is there any difference between their mechanism?
That's what a mechanism is different. The common in both is light arresting complexes getting phosphorylated. That is the common. So here light is a signal, correct, inducing the state transitions in light induced state transition. In the dark induced state transitions, cyclic electron transport is circulating, where it is getting circulated by dark cycle, that is the metabolic cycle. Through metabolism, I think maybe I can show you the data. Oh yeah, here. So you can see here, through glycolysis, I didn't show you the glycolysis or metabolic pathway. From that NADPH is getting, have you heard pentose phosphate pathway? Pentose phosphate pathway produces lot of NADPH, oxidized, when it get glucose is oxidized, you get NADPH. These NADPH is required, it goes, interact with PQ pool, then it will be circulated. Instead of coming like this through light, these NADPH are very good question. These NADPH are important in dark cycle. That is the signal.
Sir, it's a basic question, but I don't know the differences in between NADPH and
Okay, that is a metabolism. NADPH is, both are same. Here NADH, there is no phosphorylation.
NADPH, but when we are talking about glycolysis, then we see the production of NADH. So I don't know.
Okay, NADPH is not a, NADPH is, it's a reduced, when PQ pool is, when the photosystems get, the final end products are NADPH and the ATP. So NADPH will not give any ATP molecules. It is a oxidant, it forms a redox oxidation mechanism, where they reduce, it requires NADPH for a Calvin-Benzil cycle to synthesize. So it will not go to mitochondria. So here, mitochondria produces lot of NADPH. When it breaks down glucose molecules, it produces energy compound, that is the NADPH, NADH. This NADH is going to the respiratory cycle, where mitochondrial complex, where it generated NADH to ATP molecule. The ATP which we get in most of the animal system, or in the plant system, that is through NADH. NADPH is coming to synthesize, in the plant, the Calvin-Benzil cycle it goes and interact with the CO2 and then goes to the Calvin-Benzil cycle to get the sugar molecule. However, the NADH, in the animal system also have a NADPH without photosynthesis. That is through the pentose phosphate pathway, and that is not producing ATP. But NADPH is oxidant is required for reducing the oxidation reaction. Anything like there is a stress mechanism ROS produces. This goes and react with the glutathione transferase. Glutathione is the one, it also antioxidants, where it will be playing, NADPH will be playing. NADPH will not, let's take example, NADPH will not produce any ATP, NADH produce ATP. Clear to you? All in chloroplast one. It's only chloroplast. Chloroplast again, chloroplast and thylakoid. Chloroplast is a big structure. I'm just globally I'm telling. and inside chloroplast, in the thylakoid it happens. Everything is in the thylakoid. But you would say the chloroplast, this is the chloroplast structure. I don't know, there is also the metabolic pathway is happening in the chloroplast. That's why I said chloroplast. Yes.
Sir, actually in winter season the plant does not have leaves, sir. So that time in that they are using ATP, the life cycle will be going on actually. So ATP will be after the completion of ATP. I mean by using up some metabolic activities occur in plants, sir. So how can they survive long days?
Yeah, that's what it has a lot of if you are doing dieting, what happens? What happens to you? I'm not eating much. I'm not eating much, and you lose weight. Plants also lose weight. So because they metabolically they using carbohydrates or lipids, they get metabolized. That system is active. That means there is enough ATP is producing. And this is the example. The NADPH will be produced, and it goes to, this is the one is happening in the higher now the state transition. This is the exact mechanism it applies to the in cold conditions. This metabolically they degrade metabolism, then through that NADPH comes and it operates, and ATP will be produced through the cyclic electron transport. The enough ATP there, they get synthesized. The dark cycle again will be there, Kelvin-Benson cycle through NADPH will be through the cyclic electron transport it happens. Clear? Not clear? Yeah, right. Thank you. Now I think one more talk on Monday we'll discuss. Monday right? Yeah, not Monday. Next, mine is on 15th. Yeah, Yusuf, you can start. Now I want to introduce my PhD student, Dr. Yusuf. Many of you know, right? Yesterday you had a demonstration, right? So Yusuf did PhD under me with anaerobic bacterial photosynthetic organisms. That is a Rhodobacter alkali tolerant. So which grows in high salt, which grows in high pH conditions, where you have observed several kind of light arresting complex, change in light arresting complexes, which he is going to discuss. And he did Masters and PhD from University of Hyderabad. He is an Inspire Fellow throughout his career. With this, he is a good student. I asked him to present because he is a good speaker. Come.
For your kind introduction. And so, good afternoon and one and all. I'm audible, fine. Okay. So today till now we have you have gone a lot of studies on eukaryotic photosystems like plant and algal photosystems, and so as I worked on photosynthetic bacteria, which is as you all know, photosynthetic bacteria is not only cyanobacteria but there are some other bacteria also which is like purple non-sulfur bacteria, anaerobic photosynthetic bacteria, and purple sulfur bacteria are also there. Okay, so I will take you through one of these bacteria, which is known as Rhodobacter, and it comes under the class alpha proteobacteria, and they are anoxygenic, but they are not. It's not like that they cannot tolerate oxygen, but they like to grow in absence of oxygen, whereas in presence of oxygen they feel kind of stress, and they do not form the photosynthetic apparatus. So title of the light harvesting antenna, photosystem 1, bacterial organisms and algae. So let us see, when we first see the absorption spectrum and the electromagnetic radiation, when we see, is it working, this one? Okay, from here. So when we see at the, no, okay, fine. So let us see at the electromagnetic radiation. So what happens, the energy which comes on the surface on the earth is usually from 400 to, it is encompassing UV rays and everything to the far red and radio waves till 1400 nanometer, right? So some part of it which is already absorbed by the eukaryotic photosystems, right? But and eukaryotic photosystems, eukaryotic photosystems are usually able to absorb from above 700, like maybe let's say till 750. After that there is no competition. So what has happened in somehow in nature, these bacteria, where although the energy is very less in these wavelengths, right, because the number of wavelengths, number of waves is lesser, is less in the case of the higher wavelength. Okay, so energy is less, but the quantum or the abundancy is more. So somehow these bacteria have evolved not to compete with the plants, but somehow their ecological niche has also kind of let them evolve, so that they can go to not disturb the plants and other. Let us go for the higher radiation, higher wavelengths. Okay, so in that way you can see there is many species like Rhodopseudomonas rubrum, Blastochloris viridis, Rhodobacter capsulatus, and there are many other organisms and the photosynthetic bacteria which harvest till 1100 nanometer. Okay, you can see here one of the species is Blastochloris viridis, which has bacterial chlorophyll B, bacterial chlorophyll A containing, and there are many other types species of the bacterial chlorophyll A, like A, B, G, E is also there. Okay, so these bacterial chlorophylls, when we compare with the plant chlorophyll, you can see the structure of the plant bacterial chlorophyll here. They are relatively more saturated. Okay, the first and second ring of the tetrapyrrole ring is quite saturated. Compared to that, when you find it is not quite, it is relatively more saturated, and so you can so this somehow helps these photosynthetic organisms to absorb the far red region of the electromagnetic radiation. So this is the first component actually of the photosystems. To talk anything, first you should talk about the bacterial chlorophyll. Whereas the tetrapyrrole ring remains same actually, like phytyl chain almost remains the same. So when we see the comparative point of view, if you see what kind of electron transfer, photosynthetic electron transfer types are there in the nature, so you can see the Q type of reaction center and iron sulfur type of the reaction center. Under the Q type of reaction centers comes the oxygenic phototrophs and purple bacteria. So here in purple bacteria, this is the one of the anoxygenic photosynthetic bacteria, which has a very simple electron transport chain. And when you see that this P870 is one of the, it present bacterial chlorophyll A dimer, which is present in the reaction center. from where the charge separation occurs. Okay. So from charge separation, it goes to bacterial pheophytin. This is excited state, and then bacterial chlorophyll excited state, bacterial pheophytin, then quinone. And so bacteria doesn't have the plant kind of quinone. It has the ubiquinone 10 or menaquinone. Okay. So and then from here it goes to QA, QB, QP, and then cytochrome BC1 complex. So bacteria doesn't have cytochrome B6F complex. It is less advanced, less complex actually. It is very simple, and these are the component subunits of the cytochrome BC1 complex. Although some of them are similar to the plant photosystem. Okay, plant cytochrome B6F complex. And then again the electron from here goes back to reaction center by the help of cytochrome cytochrome C2, which is a mobile element. Like, okay, it is it can solubilize, it can like mobile form in the, it can pass from the membrane. It is present, cytochrome BC1 is present in the plasma membrane, right, membrane lipid bilayer, and then from there it takes that electron in the lumen, it remains in the lumen, and from there it again goes to the reaction center. So likewise, oxygenic photosynthesis you already know, so it is containing both Q type of reaction center and iron sulfur kind of reaction centers. So PS1 and PS2 already everyone has talked earlier, and then green sulfur bacteria have the iron sulfur kind of, so only one photosystem is working in this kind of in these bacteria, whether only green iron sulfur type or the quinone type of the reaction centers. Okay. So moving to next, so I'll be focusing on this kind of quinone type of the reaction center, which is the representative species which is extensively studied is Rhodobacter sphaeroides. So let us see how the photosynthesis occurs in these bacteria. So here is the structure of intracytoplasmic membranes. So in case of the plants it is thylakoids, but here you can see it is the intracytoplasmic membranes. It is a spherical structure and which contains the light harvesting antenna and known as the LH2 and LH1. So these green ones are known as the LH2 and this blue one is called the LH1, and these LH1 actually contain the reaction center. So when you see the structure of purple photosynthetic bacteria, you can see these are the intracytoplasmic membranes which are formed here, and then this is the structure, and this is the LH2. This is kind of LH1, it is present in the dimeric form. So it depends upon sometimes upon the physiology of the bacteria, that physiological condition, that sometimes this dimeric form is present in the form of the monomer or sometimes in the form of the dimer. Okay, so when we see, you can see this one. I think so. It is not one. Yeah, sorry. So circularly you can see, and usually it may have 1 or 2 ATP per ICM, intracytoplasmic membrane, you can say this. So, and when we see the cross section of it, you can very easily see the light harvesting antenna 2 and how the reaction happens. This is LH2, LH2 transfers energy to LH1, and then LH1 transfer energy to reaction center, and from reaction center by the help of the quinone. Quinone here it is ubiquinone. Ubiquinone actually from here it transfers, it like moves within the lipid bilayer and then reaches to cytochrome BC1 complex. Cytochrome C2 is already attached at the luminal side or the cytoplasmic side, you can say, and this is the periplasmic side. Okay, like here in this case it is periplasmic inverse. Okay, this is remaining towards the luminal cytoplasmic phase. So and then it moves to reaction center. So let us see on the structure of the LH2. Usually LH1 and LH1 remains bigger in size. Here LH1 remains bigger in size, and it is made up of the 2 heterodimeric membrane protein complexes known as the LH1 alpha and LH1 beta. Beta is this one, LH1 alpha is this one, and usually the number of the LH1 and LH2 remains around 13 to 14, and some species varies, but not more than 15 or something like that. Okay, and it encompasses the reaction center. Reaction center is made up of the very simple reaction center. 3 reaction center proteins are there, H, M and L, heavy, medium and lightweight. Depending upon their molecular weight, they have been labeled as the H, M and L. And they also have another protein known as the Puffx protein, which actually makes a nick. In the nick means it cuts the circle so that ubiquinone can come and reach, can have the access of the reaction center, and then go to get reduced and then reach to again go to cytochrome B7 complex. Okay, so this is LH2. LH2's work is nothing but harvest the energy and then transfer to the LH1, and from LH1 it again goes to the reaction center. So LH2 is relatively smaller in size, and it is also made up of the alpha beta heterodimers. This is alpha is the inner protein and beta is the outer protein circle. Okay, and these are very small, 8 to 9 kilodalton proteins, and so this is the transverse section of, this is ICM lumen, you can see. So what happens, protons get accumulated inside here, and then this is used by ATP, again proton is accumulated here, and this is used by ATP synthase to produce ATP. So as such, in bacteria this is very simple, and its work is only to produce ATP for the expenditure. So when we see the crystal structure of the photosynthetic bacteria, which has been, this is the cryo-EM structure of the photosynthetic bacteria from Rhodobacter sphaerides. We see that dimeric protein reaction center complex, cytoplasmic side, and this is the cytoplasmic view we are seeing from the cytoplasmic side, and you can see two dimeric, one dimer is there, and it is containing this Puffx protein. Two Puffx proteins are there, and how they are interacting. So this Puffx protein has a is nearly 82 amino acid, 8.3 kDa protein, nearly a very small protein, and it interacts with, you can see the interaction, reaction center H, protein, tyrosine, alanine. Alanine is from alpha, and then this is from arginine and glycine, lysine of Puffx. Puffx is red color, so the one which are labeled in red amino acids, they are interacting with the reaction center H and tyrosine and arginine. So these are the interacting partners which make the dimeric form. Okay. So likewise, you can also see here the C terminus, like periplasmic side. This was the periplasmic side. So when we just see how this at this periplasmic side, how the interaction is happening here at one side. So here these are the different amino acids playing important role in the interaction, but these interactions are not covalent. These are hydrophobic, otherwise the salt bridges. So mostly hydrogen bonds are hydrophobic interactions. So then there is another membrane protein which is present actually when we check the different species. So we find that some species do not have the Puffx, they have some other protein called the W protein actually. So that W protein is not this much tilted, okay, and it does not make that much bigger nick as we get here, but it makes a smaller nick, but although it doesn't make the dimer. Okay, it is from Rhodopseudomonas palustris. In that, W protein is there, known as the W protein. That W protein tries to make a nick which helps for the quinone to come and get reduced, but it doesn't help in formation of the dimer formation. Okay, so somehow the physiology of that bacteria is bit different. So that is what I meant to say. When you examine different kind of photosynthetic bacteria, their physiology actually, because of their physiology and genetic makeup, their photosystem structure is different. So let us go PUF-Y, another protein was earlier it was not identified, this PUF-Y protein. Why? Because this PUF-Y protein was getting masked when they were running the SDS page analysis. This PUF-Y protein was co-migrating with alpha protein, alpha and beta protein, because almost there is a very less difference in the molecular weight of alpha beta protein, 8 to 9 kDa, and that protein actually this also has 5.8 kDa protein. So somehow it was not identified till now, till the paper, this paper was published. And so let us see how this protein is looking like. So it actually it has both N and C terminus are oriented towards, present towards the cytoplasmic side only, but towards the periplasm it makes a loop, and its interactions you can see here, like these amino acids, how they are involved. Okay, but specifically it doesn't interact with all the alpha beta proteins. It especially interacts with LH1-13 alpha and LH1-14 alpha proteins of light harvesting antenna 1. Okay. So there was a lot of study on this, and actually there are many different classes of light harvesting antenna 1 and reaction center, I mean dimeric reaction center classes will get made in the biogenesis of the photosynthetic apparatus. So what happens, many classes like 2 classes were identified in this photosynthetic bacteria. It was their orientation, their point of interaction, like here class 2 and class 1. Class 2 has some tilted orientation, like these two are the LH1 reaction center LH1s, they were interacting somewhat like this. So the angle here formed was nearly 160 or 148, so it was different. So what they proposed, that in the assembly of the reaction center dimeric form, what happens, first the reaction center is there, get assembled, and then after that the PUF-X protein which comes and interact with the H protein. We have found the H protein, it interacts with the H protein and then goes, then subsequently LH1 proteins get assembled. After that it gets completed, and then comes the PUF-Y protein, gets inserted in the structure, and then recruitment of the terminal LH1 subunit. So LH1 subunit gets comes very in the very last, and then at the same, then after that when dimer formation occurs, so since there is a lot of interaction happening, right, like hydrophobic or salt bridges interactions are happening. So because of that, what is happening, so it takes some time to reach the final stage. Okay, because of these interactions, because it is a big molecular complex, protein complex is there. So this is an intermediary class which gets formed, and later on structural remodeling, by the structural remodeling it gets into class 1, which is the final class of dimeric form in case of the Rhodobacter spheroides. Okay. So after this, in this study, what they did, after all the crystallographic analysis, they found that what could be the way of quinone, how it comes inside and then how it interacts. So they also did the RMSD simulation, and to find out how they behave in silico, in vitro, how they would be interacting, like how these molecules interact. Okay, so they did, and after that what they found, that during the simulation study they found that Q3 tends to move towards QB. So this is the reaction center, you already know. Reaction center has the 2 dimeric bacterial chlorophyll, and then after that QA, PA and PB, 2 dimeric reaction center, dimeric chlorophyll present in the reaction center of bacteria known as PA and PB. Those PA and PB after that goes to QA and QB, these are the quinones. Okay, so quinones and QA and QB. So what happens after RMSD simulation, like they got this structure, but they were not able to see how the quinone, like, so they did simulation tells like suppose if you take a medicine, paracetamol or something you drug. So there you also people do the simulation. So how the drug efficacy would be there? Efficacy in the sense how in your body drug is going to be, like where it will be going, how would be, what would be the fate of it, and how it would be interacting, where it would be going. So by simulation people try to depict that, I mean interpret or like kind of see how it will be behaving. So what they found that Q3 tends to move towards QB and QI tends to move towards Q3. Being on their dipole movement is somewhat like that it moves towards the QB and QI moves towards Q3. So in that way, so quinone can come here and interact with the QI, then Q3 and then QB, and then finally move back to go to reaction center and then get reduced and then try to reach to the cytochrome C2, cytochrome BC1 complex, right? So in this way, the reaction and charge separation, after that like electron transfer occurs. So this was the reaction center antenna, reaction center LH1 antenna, and this now LH2. LH2's function is nothing but just to harvest the energy and transfer it to the reaction center by the help of intermediary complex LH1. Okay. So this is the crystal structure of it, and this one is without protein, and this is the cytoplasmic view, and you can see how. So there are two kind of the bacterial chlorophylls. See here there is only one bacterial chlorophyll A, which is able to absorb at 800 nanometer and 850 nanometer. At the same time, it is also absorbing in the carotenoid region. So it is not competing with the plants. Okay, it is taking the far red region, that is 800 nanometer and 8Y, and these are known as the QI region of the spectrum. Whereas this is known as the Soret region. This this is known as the Soret region. So you can see why it could be, because bacterial chlorophyll, how it is present inside, it's about its orientation. Maybe the orientation is the one which is causing this bacterial chlorophyll A, being the same species, able to absorb at the different wavelengths. Okay, so it is the orientation here. Bacterial chlorophyll A 800 is present in this form, whereas bacterial chlorophyll A only 800 is absorbing, is present in this form. This is the carotenoid, which is in parallel to the alpha beta proteins, like alpha beta proteins are standing, carotenoid is also standing here having its interaction, but bacterial chlorophyll A1 is like this, somehow arranged, another one is like this. Okay, so this is what is the arrangement system or nature has made. No, it's not opposite to each other, it's kind of perpendicular, you can say. Only one chlorophyll. I mean, it's one species only, but two copies. So, sorry. So further, when we see the pigment protein interaction of B800, so because it also has to get stabilized, it is very compact structure, and it has to maintain its I mean morphology, and because for the electron transfer or the charge transfer, like by resonance energy transfer or something, forced transfer mechanism is there by dipole movement of the chlorophylls and carotenoids. So in that, it has to be maintained. The distance between the molecules is very important, and that will be the important thing to for the molecule, for the charge to move forward and move in a very specific way, very specific speed. Okay. So here you can see this is the interaction of bacterial chlorophyll A, which is absorbing at 800 nanometer, and this is the so it is this is the distance between bacterial chlorophyll 800 B800, and it is interacting with beta protein, arginine and histidine. Okay. So here, and this is asparagine here, this N3, it is so these are the distances they are making. How you can see after the crystal structure, you can see how the distance, how much is the distance, which is like how they are arranged in the photosystem, you can see. And this is bacterial chlorophyll B850, which is interacting with histidine, serine, tyrosine molecule. Okay, so these are the interacting partners coming from the bacterial chlorophyll proteins, alpha beta proteins, and the bacterial chlorophyll A itself. Further, so it is now necessary to know what is the mechanism of transfer. So we have seen that bacterial chlorophyll B800 is like kind of present in a membrane plane, whereas it is parallel, it is perpendicular, the other one B850. So the energy, what happens first, B800, which is more energetic, 800, 850 or 875 or P870. So B800 has the more energy, right? Why? Because it is more shifted towards the left side or high energy wavelength, right? Understood? Okay. So B800, which is having the highest energy, it transfers its energy to B850, okay, because it has a less energy in comparison to this. And then from here, so what happens, there are so many LH2 and LH1s, and like this will be dispersed, so many LH1 LH2s are there, okay. And sometimes there will be stoichiometry also, there will be some pattern also. So then energy gets transferred from either it goes to another LH2 subsequent or neighboring one. If it is not there, if suppose some reaction center itself is there, so it will directly transfer here to here. Otherwise, if it is not there, reaction center is not there, it will try to give it to another neighboring one will transfer to the reaction center finally. These are the time 1.2 picosecond is being taken from 800 to 850, and then nearly 5 picosecond from F850 to here B875 nanometer. And then from so here there is a difference, you know, P870 is the charge separation bacterial chlorophyll dimer present in the reaction center. So from B875 it has to go to P870, and it is taking a bit more time, 30 to 50 nearly 30 to 50, 35 to 50 picoseconds it takes to transfer. Why? Because the distance is more here also, and there is a purpose of this for because why it has been done like this, because once the energy comes here, it has to pass here. So once it has passed, and because the distance is more, suppose it has started, it got the energy and it started passing and it got, okay, and it will, the reaction center will start charge separation and subsequent quinone and the transfer will occur, but that also takes some time. So this distance actually, so if distance is not there, this will keep getting excited and will keep getting transferring the energy to it. So that will get again from energy transfer from here to here is known as the actually trapping. From here to B875 to reaction center is known as the trapping. It is not known as the transfer. It is known as the trapping. Transfer is happening all over here, but here it is transfer from light harvesting antenna B875 to reaction center is known as the trapping. So this trapping, so if it is not far, so detrapping will be more possible, okay. So in that way, and also it will be possible that they will all getting piled up here if the distance is not less, okay. So that is somehow the purpose. In that way it has been designed by the nature that the system occurs like this, okay. And the mechanism which has been proposed for is the Foster transfer mechanism. It is a non-radiative resonance energy transfer through the coupling between the transition dipoles. Transition dipoles is the like in the tetrapyrrole ring, and also in the carotenoid region, carotenoids you can found the, because they are the, when there is energy transfer they are in excited states, so dipole movement is there, the main thing, right. So further, so till now, what happened? This bacteria can, Rhodobacter can, Sphaeroides can absorb till 875 nanometer only. But there are another species like Rhodobacter blastochloris viridis, not Rhodobacter, blastochloris viridis, which can absorb till 1100 nanometer. Okay, so why is that so? So nature here in this case has given another subunit known as the gamma subunit to the blastochloris viridis, which actually rearranges the system in such a way, means the bacterial chlorophyll in such a way that it is able to absorb till 1100 nanometer as well. It is because of the gamma protein. So you can see the structure and everything is same. Reaction center M, L and H are already there, and alpha beta proteins are there, and out of the alpha beta protein, then gamma proteins have also come here. Okay, so in this way, what is happening? The rearrangement in the in this sandwich, actually circular sandwich, the rearrangement is changed. So that is leading to the absorption at more higher wavelength. So further, what they did actually, this alpha beta, this gamma protein actually is encoded by 3 operons. Okay, 1, 2, 3 and 4, known as they have labeled is gamma 1, 2, 4. Fourth is actually they have taken a pyralog of it from another bacteria. So what they did, 1, 2, 3 they deleted, and then 4 they have given. So what they are, you can see here, the wild type is absorbing somewhere till here. Yeah, sorry, there it is maximum absorption is till 1018, not 1100. Blastochloris viridis 1018 nanometer. Okay, actually 1100 it goes till here. This is also absorption, but the peak comes at 1018 nanometer, maximum absorption here. But it is not like this, these energies are not getting utilized. This is also getting utilized. Okay, so wild type is having this, but when they mutated gamma 1 to 4, so it has again come back to 950, around like 975 here. So when they are again complementing it by the LH1 gamma, so they found it has again shifted. So it is the stoichiometric rearrangement happening within the system, within the proteins, that is leading to the more shift towards the far red region. So here this is the Rhodobacter sphaeroides I grew, and this is anaerobically how I am growing this bacteria. I am not giving a single space to it to absorb, to have oxygen. So if oxygen it gets converted completely in reddish form, and here you can see completely anaerobe is there. So what I say, so there should be one question that like oxygen is also present in the media which I have given already here, right? Because it is also present in the dissolved form. So what do I do with that? Because that may also will cause the trouble for some time, because it will elongate the growth. Bacteria won't be able to adapt that easily. So what I did, usually I just inoculate the bacteria and then leave it in dark for some time, nearly 1.5 hours, and let the bacteria just like that sitting and doing the respiration. Okay, so by the respiration it will take all the oxygen dissolved in the media, which while preparing it might have taken all the oxygen, and then I expose to light, then it starts its anaerobic photosynthesis. So then after this I grew the bacteria. This is the TEMI structure. So these are the intracytoplasmic membranes. You can do sonication. After sonication, and I made the gradient 15 to 40% gradient and then loaded on it. So this is a special bacterial intracytoplasmic membranes. Okay, I stored, and then after that further experiments can be done. So further, these protein super complexes actually, which we talked like dimeric reaction center, monomeric reaction centers, they can be separated and visualized by BN page also without denaturing them. BN page blue native is because the color blue, a dye which is used here. So that's why unnative page means there is no denaturation. Okay, as it is, the protein is going to be run, and then in what form, what complexes, because denaturing causes the detachment of the protein. Denaturing means the protein is not going to be in its 3D conformation, right? So here you can see the green gel, completely green complexes. You can see all these complexes, reaction center LH1 dimer, and then ATP synthase is not visible here. You can see ATP synthase has also come, but it is not visible. Why? Because ATP synthase as such doesn't have any pigments. Okay, but after staining I have got this ATP synthase as well. So you can see this is dimer. Dimer has the more molecular weight, so it came up, and then monomers, and then LH2 major and minor are also their species. So that can be, that has been separated. So in that way you can do. And further, if you want to, I wanted to go for identification, because here what is this protein which is coming after the after staining. So I wanted to identify, because I didn't have an antibody for this, because bacterial ATP synthase is bit different from the algal cultures. Okay, algal ATP synthase. So and then the 2D I did, and then I gave these protein bands. I just picked it up and gave for identification. So found 4 subunits I gave, and found it to be alpha, beta and gamma subunits having the molecular weight of 55 to 19. Okay. So like this you can do, and then you can also separate these complexes by the help of sucrose density gradient. And so for this, what you have to do, actually you have to solubilize the super complexes. Actually you cannot directly load after isolation. You have to solubilize the complexes, because these complexes are what kind of protein they are? Hydrophobic or hydrophilic? They are hydrophobic proteins. Okay, so they are present in the lipid bilayer. Okay, because they are the hydrophobic protein, they are membranous proteins, so they need a hydrophobic nature environment. So in order to take them out from the thylakoid, from the intracytoplasmic membranes, actually not thylakoids, so intracytoplasmic membranes, I have to provide another detergent, another environment which can mimic, which can give it a house from where it can shift. So I have, you can dissolve it in solubilize it in beta DM or alpha DM or some other hydrophobic detergents, carbohydrates, polymers also. So usually I have used beta DM, endodecyl beta D maltoside, which gives the hydrophobic environment and it dissolves. So then I separated by sucrose density gradient. So here you can see I got the LH2, LH1 reaction center monomer and dimer. Monomer and dimer you remember, like earlier I told, right? So then I did the absorption spectra, and you can see the absorption spectra here. Like I am getting the 800 nanometer and 850 nanometer bacterial chlorophyll, which we have already seen that the absorption, it absorbs at 800, B800 and B850, and then this carotenoid region, Soret region. And then when you see that dimer has a more density, right? Because it has a more weight. Okay, so it comes in the lower phase, and this lower phase you can just when you can see here the dimeric. So the reaction center phase, it shows that absorption at 875 nanometer. You can see here that it shows that here in these cases dimeric form is present and it is containing the reaction center, because the B875 which absorbs at bacterial chlorophyll B875 is present in this, not in LH2 fractions, because LH2 contains only B800 and B850, right? Whereas LH1 reaction center contains the B875, but LH1 reaction center also contains the bacterial chlorophyll dimeric reaction center, that is in the center where the charge separation occurs, that is B870. So here one can ask the question, why the B870 is not coming? Okay, because B870, it can be masked by the presence of B875, and that too B875 is present in a larger proportion. For B870, only 2 species of 2 molecules of bacterial chlorophyll are there, right? Whereas for the LH1 reaction center, what you might have seen in the previous slide, that there are many of the species of the bacterial chlorophyll B875. So when you run the again SDS page, I did this silver staining, and then you can see 3 proteins only. These are cytochrome BC1 complex protein. Sorry. These are cytochrome BC1 complex protein, and this is reaction center proteins, and probably this is some also cytochrome BC1 complex protein only, but it is in very minor concentration, and here alpha beta protein, and also probably puff-Y may be here, but I'm not very sure because the resolution is not very good here. Okay, so like this, so you can also see, like we have already seen, there is a pigment protein interaction, because pigment proteins has to be there, and because it has to be in a compact form, right? So what interaction? So if you have to assess the interaction, how can you assess that interaction? So there is one technique called as circular dichroism, where we use the circularly polarized light. You know polarization, circular polarization, and that you polarization nature of light, that you can tame it according to you. It's a polarization, you can make it according to your wish. Okay, so it's components, they are traveling in a circular motion, circular way. So in that light, if passes through it, so we can so through that we try to assess the interaction between these pigments and proteins. Okay, so if it is loosened somehow, if it is loosened, so what will happen? The interaction peak intensity will go down. Okay, so here in case of the control condition, like high pH condition where the interaction is too much, like too good, so there the intensity is high, and whereas in normal pH the number is also less. At the same time, interaction may also be, sometimes it is also concentration dependent also. Concentration dependent, not in the sense like you can say interaction only, because here you take at equal chlorophyll, whatever the experiments you do, you take at, always do at a same platform. Whatever the experiments, even you have to do in science, you have to give the same platform for everything, whether equal chlorophyll, equal carotenoid or anything. So this is another way. So when we look at the intracytoplasmic membranes, how are they formed actually? So what happens when the bacteria gets divided? Obviously it gets some of the intracytoplasmic membranes by its mother cell, but when the cell elongates its size or something like that, when it matures, it has to make some intracytoplasmic membranes, and which gets formed by the invagination, membrane invagination, plasma membrane invagination, and later on it gets detached from here. So there are some proteins which play important role. Suppose in case of the purple sulfur bacteria, this is purple non-sulfur bacteria which I am talking about, Rhodobacter. Okay, in case of the purple non-sulfur bacteria, there are some proteins known as FMO protein, phenylmethyl-olsen protein, which helps in formation of this protrusion. Okay, this invagination, it helps in form. Whereas in this case, in case of the Rhodobacter spheroides or Rhodobacter species, it is the mixed cysti protein which tries to somewhere get here located, and it helps in the formation of the this intracytoplasmic membranes invaginations. Okay. No. Yeah. And when you see this structure, so this is atomic force microscopy images. Atomic force microscopy is another tapping method of membranes. By tapping method, it will go on the surface of the membrane. Suppose you take out some membrane, plasma membrane, and you want to see the structure or the face of its surface. Suppose this is the membrane I want to see. So the atomic force microscopy will try to give you the surface structure of it, how smooth it is, or is there some protrusion is there, or something is bulging out here and there, or if it is having some shapes here. So that is what they have taken out here. The people have taken out the intercytoplasmic membranes. Sometimes they give mild detergent, otherwise they can go directly ICMs you take and it will be visible like this. So here you can see these are the LH2s which are present in sometimes pentameric form or hexagonic form, and this is the dimer. How it is just in vivo you can see, actually just take out the intercytoplasmic membranes and go for the determine some very less concentration so that it is not very jumbled over there, just very less concentration, so there is no overlapping of the proteins, and then you can visualize it. So these where there is lights coming out na, those are the reaction center, those are the containing the reaction center, light harvesting antenna one containing the reaction centers also. Okay, so in this way you can see, and even you can visualize the morphology of it and how it is looking like, and sometimes if you give some treatment or some conditions you apply, how the physiology or the morphology will be changing, you can detect it by atomic force microscopy also and by TEM imaging also. I think I have TEM image. Yeah, I have TEM image also. So this I did the transmission electron microscopy for my cells, and then here also I found this ICMs present here, and also you can see one more phenomena, actually the bacterial physiology and the shape is also changed, right? You can see something here. So it is because of the high pH grown cells. High pH is actually causing cells not to divide. Okay, so I used the cells gave three conditions, like 3 light intensities, 30, 250 and 500, where in case of the normal pH conditions like neutral, around neutral 6.8 pH, so what happened, the bacterial cells size starts like kind of reduced, and also obviously if the cell size is reduced, the ICM number is also reduced. So what happened here, so later on after the analysis of omics and omics analysis, I found that the cell division assembly is quite fast. I mean cell division is quite faster in this case. Even I was quite confirmed from the from growth curve itself only, from generation time was quite faster in case of the highlight conditions. But I was like after the TEM analysis came, so from the TEM analysis it was quite visible that bacteria is dividing so fast, and highlight is causing actually the bacteria to divide quite fast. And from transcriptome analysis I found that the assembly the septum formation, cell wall formation, like if bacteria has to divide, there has to be some proteins, right? Some proteins, the septum formation proteins are there, they have to assemble. Okay, so like FTSZ protein is there, right? FTSZ protein is the one which is the major protein for the assembly of the septum formation, and so its expression was too high in this case, whereas it was very less because this high pH retards the assembly of the cell division proteins. So although effect of highlight was there, I found the effect of highlight here, the cell shape is quite elongated, but it is decreasing with time, with increase of light. With increase of light the size is decreasing, but its extent is not that much. Okay, so in this way, so if I would have gone, I tried to do atomic force microscopies, but somehow good images I didn't get. So but one could see for the this also how the physiology of the this how the surface or the face looks like for the intracytoplasmic membranes. So this is all, and about this rhodobacter, and being very simple, but it has a it can cope a lot of it can grow till pH 10, pH 11. It has its physiology is quite strong and it can cope. So that's all. If anything is there, you let me know. Any questions? Okay. It feels like you got it, everything. If there is nothing, then we can go for lunch. Okay. Thank you one and all.
Fine. Switch over challenge. It came out. See? Fixed up. Rather than holding my hand and speak. Just for they are taking maximum time. Yeah, we can exchange. Whoever is speaking.
Yes, sir. Okay. You want to proceed?
This,
See,
delete
take
this.
once.
Delete this slide.
Yes, sir.
This slide. NPT. NPT. Yeah, fine. Just save it. Mike, I'm keeping it here.
Hello, hello, hello. आवाज तो आ रही है. अरे
So you can club into a group of 4. You can club into a group of 4. So yeah. So everyone are there, no? Let's go into the program. So today, have you all gone through the what are the program, what are the experiments we are going to do? Okay. RNA, mostly RNA, and one more thing will be followed by if you want to isolate a particular gene from the entire genome, how you are going to do, followed by, yeah, you can give me a pointer. So this is the entire program outline. So we will be having 2 days of molecular biology experiment. So today will be the first day. So in first day we will be dealing with the RNA isolation from Chlamydomonas reinhardti, followed by using the trisol method, and next will be target gene. So you can implement the same colony PCR, any gene using the same method you can isolate, and further followed by the next day, the second day will be on Tuesday. So that day we will be cloning the gene into a vector and we will do the algal transformation. So that will be the entire molecular biology structure. So anyone repeating the course, like last year also we had the same almost similar program. So any of you? Yeah, not about you, no. Okay, fine. Most of you are new. So how familiar are you to the RNA isolation or whatever the experiments mentioned here? Namrata, can you help? Yeah, I just asked how familiar anybody has done these experiments earlier, so that we can little bit faster. Kit method you did. So trisal you haven't. So you're already in advanced. Yeah. So what's your name?
Nandana.
Okay, maybe camera is here. You can come forward so that, yeah, you can come forward. Sorry. Yeah.
So you have done already.
Yeah,
Okay.
RNA. Yeah, I have done the RNA isolation, cDNA preparation,
Okay.
qRT-PCR, that things.
So already you have done. You are from which university?
I studied masters in Pondicherry.
Oh, Pondicherry University. Okay, nice. So any of you other than her? So masters, you are all from masters, right? So nothing RNA, no isolation, DNA isolation, nothing. So maybe it will be more informative and interesting for you. For you, it will be whoever has done, it will be a repetition. So you said you done with the kid, right? So kid is always advanced, and particularly when you go for starting a new lab, obviously you should have some basic knowledge so that you can save money. So trizol has a both advantage in a way that it will give higher concentration. The yield will be very high, and it's a bit laborious, but still the economic point of view, it's going to be beneficial. So why don't we try that? So that's why I kept the trizol method. So any doubt in the program outline, you can ask me now before going into the experiment. If you have any doubt or if you have some questions regarding the experiment, anyway, she will be explaining you about the what are the components and how the experiments will be done. But before getting into the experiments, any doubt, any clarification from you all, you can ask me. Yeah, it will take. That's what we start early so that we can complete early. So we will be simultaneously doing the both. So because trizol method is a bit time taking, so anyway you will be having incubation times and all will be there. So in that time we will club the colony PCR also. So okay, let's move into the experiment. Okay, so now Namrata, she is my lab member, so she will be explaining about the trizol method.
Thank you, sir. Hello everyone, myself Namrita. So we'll be discussing about RNA isolation by trizol method today. So before we proceed with the experiment, you guys know why do we do RNA isolation? What is exactly RNA isolation? Anyone? Just for cDNA preparation. Okay, I'll tell. So basically, RNA isolation is extracting intact molecules from cells or tissues or anything. So which we can use for further downstream processes like qPCR and RT-PCR, transcriptomic analysis, RNA sequencing, or Nathan blotting. And also, if you want to study any genes which are expressed under some stress, you're giving salt, temperature, so we can help study with this RNA isolation. So first before coming to the experiment, first we'll grow clammy until it reaches the OD of 0.8 to 1. And then we'll harvest 100 ml of cells, or depending on your organism, whichever you use, you can decide the concentration in which you can take. And after harvesting and collecting biomass, we take liquid nitrogen, pour it on mortar and pestle and grind, and then we can break the cell walls. So once you get the fine powder after grinding on mortar and pestle, we transfer the powder in the 1 ml of Eppendorf and add trizol. So why do we add trizol? Anyone? Yes. So trizol basically is a monobasic solution which contains phenol and guanidine isothiocyanate. So phenol helps in dissolving proteins and lipids, and this guanidine isothiocyanate helps in preventing RNA degradation. So once you add trizol, then we'll adding chloroform. So what chloroform helps in this helps in separation of mixture into 3 different layers. So once you add chloroform and you centrifuge at room temperature, you'll be getting 3 different layers. Upper layer is aqueous layer, which is RNA. The middle layer, that is interface, which is DNA, and the bottom layer is proteins and lipids. So after that, further we'll incubate and centrifuge and incubate at room temperature. And once you get the different layers, you can collect the upper layer which contains RNA. Then after adding that, after taking, you will add 5 molar of NaCl and isopropanol. So isopropanol helps in precipitating RNA, like collecting more RNA, and the NaCl helps in enhancing the precipitation by neutralizing the charges on RNA molecules. So once you add isopropanol and NaCl, we'll centrifuge it, and then we'll again discard the supernatant. So you will be getting 2 supernatant and pellet. So we'll discard the supernatant and then keep it for ethanol washing. Why do we do washing ethanol? This I think anyone can tell. Why do we use ethanol for washing? Yes, yes. To remove if there is any contamination or something, yes. So once you do ethanol washing, then we'll air dry the platelets, and then we'll elute the RNA whatever we have got with nucleus free water, and then we'll take nanodrop readings. If there is any confusion in this part why we are doing or any step, you can ask. So once you go for nanodrop readings, you will be getting two ratios. First is 260 by 280, which is generally it should be in the 2 range. So if it is beyond, if it is like more than 2.1 or it is less than 2, less than 2, like 1.5 or 1.6, then it indicates that your RNA is not pure. So if you get the 2 reading, that indicates that you have got pure RNA, which is free from any proteins or phenols. And you also have to see 260 by 230 ratio, which should be in the range of 2 to 2.2. It indicates if it is between 2.2 and 2.2 to 2.2, it indicates that there are no salts or phenols or any guanidine that you have added. So mostly the ratio should be in this range. And if suppose you don't get the ratio in this range, what thing you can do for yes? So what you can do to remove that contamination? Yes, you can wash it again. Suppose if you get the 260 by 230 ratio is not good, so you can use washing 2-3 times, which can also increase the ratio to from between 2 to 2.2. So now we'll be heading towards the experiment. We'll start RNA isolation. You will be given clammy. We grew clammy under normal light conditions in 50 micromole photons, and you'll be given 50 ml of cells. You'll harvest, and then you will grind it on water pestle. Further steps I'll be explaining meanwhile.
So how many of you worked on chlamydomonas here? No one. Oh, okay. So so what are the organisms you are working on? Plant, higher system. So bacteria, anyone bacteria? Okay. So okay, it's a the procedure will be almost similar. Anyway, no need to worry. So first step will be grinding. You need to break open the cells. Whether it's a plant or anything, you need to break open the cells. So for that, the algal system is a unicellular. So my lab members will be helping you. So as I said, we are going to divide you into a group. So that will be difficult, no? So I kept 3 workstations, 1, 2, and 3. So it will be good that you can divide among yourself into a member of 4. 12 members, no? 4 into 3, you can divide it. So make one group, group 1, group 2, group 3. So you want to be among yourself, or you can split, mix it. Okay. So you can come, group 1 can come here. Group 1, group 2 can be the next, followed by group 3 this side. Yeah. Three groups. Yeah, that side one. Yeah. Yeah, try to adjust yourself. The lab is little short, but still you can manage.
Gloves first.
Yeah.
Depending on the sizes, small and medium size are kept here. Gloves.
So just for, I have kept the instruction manual in front of you. Each group one manual I kept. Just go through the manual. So everything is written there. So we will be instructing, and you also read once and follow it very carefully. Yeah, there are gloves, 2 glove size will be there. According to your hand size, you can change it. So Shreya, are you saying something?
No, I'm just telling that.
Okay. So first step, take 50 ml culture, which our lab members are giving. Just take 50 ml culture. You are going to pull down the cells, harvest the cells. So first you are going to harvest the cells, and after harvesting you are going to grind the cells.
Previously not happening like that actually.
So everyone try to be involved so that you will be learning something. Don't be like someone will be doing, I'll be just be a spectator. So Amrita, can you shift the laptop so that we can. connection. Okay, it's already there in the GIAN group. We shared protocols were there. Just check it.
You also gave this.
Yeah, per group one we kept, but still you have in the group. You can check it in the group also. So every group collected the cells in the Falcon. Group 1 is done. What about group 2? Group 3? Okay, it's going for centrifuge now. Okay. So, and for this RNA isolation, for everyone, I'm telling it's a aseptic, sterile. It should be sterile. Whether your personal sterilization has to be done by 70% ethanol, followed by the gloves, and the entire workstation should be completely sterile. Even a small RNAs are everywhere, right? You know that, right? Even in your body, what are the secretions will give RNAs? Any idea? Saliva. Then saliva. It comes in saliva. Your sweat. Sweat also has RNAs. So saliva at least you can control your mouth, but sweat you cannot control, right? So always be careful. So sterilize yourself with 70% ethanol and completely clean the place.
Using it, yeah.
Pre-cool. I think we should.
Yes.
I think we should pre-cool the motor.
Once the harvesting, then there's a collecting 100 or 50.
Bring ice bags.
Anyone from your group?
One from each group, please follow me. We get that.
Set a group leader for your group.
One person from each group, please.
Anyone can go. Yes.
Okay. Right, similar only almost. Any kits you are following, right? The kid will have the basic principle of trisol only. They will have guanidine. Have you gone through the composition? They would have mentioned this guanidine, isothiocyanate, phenol. Everything will be there. We are doing the trisol. Yeah, trisol. Not a kit method. Not a kit. We are using trisol. You need to prepare the trisol. Trisol is already available, commercially available.
From that also, we just need, we don't need that RTRNA or we just need pure RNA. So we are doing that.
So what are the requirements for RNA? By the way, I think she asked this question. So what is the answer you gave? I was not there. I was far away. I couldn't hear it.
Now I told now they should answer.
At least now you should answer. For downstream processing. What is the downstream processing?
You guys are telling correct. Why so soft?
They are sorry. They are little scared. What if the
Even if you tell wrong, you can learn.
yeah,
Just
we are actually students only. By the way, we are not a faculty, so no need to worry. So how many are PhD scholars here in this? 1, 2, 3. So future PhD, I think so. Masters.
Anywhere you go, you have to do this. This is the basic multiplier technique for anything.
So during the COVID also RT-PCR is very popular, right? So for any basic RNA they will isolate and then they will go for the cDNA conversion. Then they will check any RT-PCR will be done with the help of a cDNA. And RNA isolation is a very tedious process. Even if you miss in one step, they will degrade. You already know, right? So it should be done in complete ice. Nowadays, kit-based methods are allowing you to do in room temperature also, but still very precaution has to be handled. Otherwise, they will degrade easily. Yeah, it has to be grinded in the liquid nitrogen. So we will be showing you first. Once they bring the ice, we put the cells for centrifugation. Once we have to remove the entire media. So almost 50 ml of cells you have started, right? So you will remove every media and resuspend it in 1 ml, hardly 1 ml media, and then you will transfer to the mortar and pestle, and then you will add liquid nitrogen and you start grinding very hard. It should be fine powder. How good you are grinding, your yield will be very high. So try to grind it very fine particles.
Fritz improve.
Yeah, each group you can keep it in their workstations. So cells are ready. Then the cells comes, no, otherwise. Good. Fine, be careful with the liquid nitrogen for the master students who are handling first time. You will get some cold burns if it mistakenly fall on your hand or something. So cautiously handle it. Fine. So at the end of the day, we will be running a gel with RNA. So if your RNA is quite intact, you should get how many bands? 3 bands you should get. So let's see at the end, yield, we'll check nanodrop, you should get 2, as well as 3 bands you should get. So let's see which group is able to bring the 3 bands and the concentration is good. Those are different RNAs are there, no? mRNA, rRNA. So those will be differentiated into 3 bands. Yeah.
Can you use the alternate ethanol for washing?
Where instead of isopropanol.
Ethanol is there.
Yeah.
Is there
Actually, it's a standardized, so we can try it. Obviously, we can try it out, but they worked actually better. So they set the protocol. So we are just following it up. So I haven't tried it. Maybe possible chances are there. We can try it out. So basically, it's going to remove all the impurities and solubilize them. Washing is solubilizing using that.
chloroform also?
Chloroform is for phase separation.
The chemical only we can use. Not there.
Yeah, means when it works, they don't think for alternative until unless necessary, right? So that's
Most cheapest, so that's why
yeah. That might also be the. So cells are ready. Harvested cells. Okay, I think these are the reagents they would have gave to you.
Yes.
Yeah, you can tell them. So
We'll open this water and pressure, and we'll pour liquid nitrogen first. Pre-cooling. That is pre-cooling.
when you go back, when you handle the things where belongs, right? Then now you open your motor and vessel, which we kept in front of your workstation for group 1. So don't keep it in
And
the
don't
floor
touch
or
it with hands. Like, not this gloves. Gloves you're wearing. Don't touch it with bare hands.
So open the silver foil and keep the motor and vessel. And where is the motor? Motor, no, they don't have the spatula. Motor is maybe one. You can use this also. I have one already. You can.
I am putting trizol here.
We are giving.
Everything is autoclave before. No need for autoclaving again.
Okay. So before pouring the liquid nitrogen, be a little cautious. It will be very cool. Pour in your hand.
Maybe we'll show and then
Yeah.
that's it.
Mask, if you have, it's more ideal. But right, it's more ideal to wear mask. So first time RNA isolations. Okay, all the best. No,
You start the supernatant. Wait, I'll provide you discover.
Yeah, don't disturb the pellet. It has to be the, yeah, keep it.
I think you can use this.
Wait for a while.
Each fruit one.
Wait for a while. Our members will come. They will explain you.
Don't give the small one.
Okay, guys, in this brown tubes is the triazole reagent. Okay, 1 ml.
Take.
Discard it, otherwise it will last.
So light side.
Pour it little fast, otherwise it starts stabilizing. See. Now you will pre-cool this by liquid nitrogen. You take.
We'll tell you.
You try.
Then you bring the chai. Because half an hour we have.
Little, right?
Yeah, very little. Not that much. And slowly. Or more, little more.
Okay, one group started. So
Yes.
carefully pre-chill the mortar and pestle first.
Nice. Give me little space. Now.
मैं इन्होंने नहीं डाला अभी तक रुको.
Yeah, first pre-cool it.
You want to pour? Then pour. Pour liquid. No. Wait. First you will pour this. Remove the spatula. Keep mortar in that. Now when you once it's cooled, then you add.
So carefully pre-cool. Once they are cool, then we shall be good. Yeah, they added already.
Yeah, right. What?
No, Shreya, you are explaining individually or to everyone, right?
Explain
Let them see first.
कौन दिखाएगा?
So you are going to every group and inform.
They asked me, so I started.
No, good. Show it one. No, it will be easy for you and for them as well.
What did you add in this?
So just come and see once she's showing what has to be done. By seeing this, you can demonstrate or you can perform in your workstation.
Come guys.
No, give a slight way for the. Come this side. This side. Come this side.
This is your
You can come this side.
So
Give some way for the camera.
Little bit, you will
Come
dissolve
this side.
first, so that you can take in pipette and keep in this mortar vessel. So first take pipette, little bit you start dissolving this after resuspending, and you should not touch pellet. Yes, be careful. Otherwise it will
Very
stick
gently
because
you
it's very
mix with a 1
concentrated.
ml pipette so that you will get
Take.
a very
This is
dark
for this is
liquid
This is
of
1
your
ml.
culture. And simply after pre-cooling your mortar and pestle, just load it, put fresh liquid nitrogen and start grinding. Fine?
This is 1 ml.
You will get enough amount of cells from 1 ml.
This is for 200 microliter. This is for 1
Just
ml.
make sure that the pellet is properly solubilized.
Open this. Take
And
little
keep
water.
the pellet
Suppose your
on ice.
whichever media you're
Always
growing, you
everything
can
on
leave in
ice.
the once you're discarding supernatant, leave it. Then you should not touch pellet directly. Entire pellet. You have to
Yeah,
suspend like
entire
this.
pellet, yes.
Okay, I'll show. Like this. Okay. Take and resuspend like this.
Any doubts?
So once you do like this, everything will be dissolved. This is
This is
water.
see, pre-cooling is
Yeah,
done.
this is
Now
water.
we pour liquid nitrogen.
If
So
you don't
now
have
it's
anything to dissolve, you can use empty water. Okay.
pellet, everything
So you have added anything into
has to be
Yes,
chilled
no,
only. If you
I'm
put
telling
anything
that
at room
I just added
temperature,
water.
your
Once you're
RNA will
discarding the
go.
supernatant, you leave just 500
So
microliter
now
of
it's cool. You can touch it
media or
and see
anything
that
so
it's
that it
cool.
can be resuspended. Because you can't take pellet directly.
Now,
Just for that.
where is the liquid nitrogen?
Now,
Now
add
this
liquid
process
nitrogen.
has to be
You have
quite
to see, once
fast.
the moment you add this, you have to
You
keep
guys,
on
did you
grinding
solubilize the
continuously.
pellet?
So I'll be adding liquid
Solubilizing,
nitrogen is
no?
there.
After that,
Pour
I'll come
little
and
bit
show you.
and then you add.
And that water should be like distilled water or milky water, anything
Anything,
specific?
empty water is fine. If you don't have media, either firstly use media, whichever you're going with. If you don't have, then empty water is fine. Now you pour a little bit more. very little. Done. Take and pour. And one person should start grinding. More forcefully. Yeah. Let them try. Done?
So as soon as we'll put this, no, this will form into a stone-like thing. But we don't have to worry. We have to crush.
More force, with more force you have to press this. The more pressure you apply, the more better RNA you will be extracting. The more fine it comes.
Okay, can you pour some more? Can someone
Sometimes
and someone needs to start grinding as soon as it. But make sure it doesn't
no.
fizzle out
That
because
the
there are
more
a lot of
force you
bubbles
apply,
coming.
the more
So
fine
this
powder
step
it
is
is, the better
very
RNA you will
tricky.
get.
Bus.
It generates more force.
Wait,
Yes.
let me pour it first. Let it
Also, when you put nitrogen, so it sticks sometimes. What happened? Need help?
Yeah, we need it.
How?
No, it just doesn't have to
Done?
be very liquidy because a
Now
lot of
you
media,
just
if you solubilize it in a lot of
You
media,
need 100
Then?
milligram of cells, so try to take as much as possible, all the cells. We need minimum 100 milligram for one reaction.
Once you add this, immediately pour this. Now take in pipette, collect everything. Now pour. Just put in this. Don't touch with pipette. Done.
okay, pour
See, it's
some.
very thick. If little bit more you should make.
It flows because of
You should always put in middle. Okay, because if it sticks now, I don't know. It's very hard to know.
put only a little, just a little.
Now you keep on grinding it like this unless it becomes powder.
Bus.
Very fine powder it should become.
Okay, just grind. Yeah. Great. Perfect. Perfect. This will give you enough fat. Okay, now when you grind it, the color has to change to ashy green. Okay, it has to lose most of the green color, and then we get a very good RNA. If it loses its green color,
So only RNA will be green color. While centrifugation, they will separate.
I think they should put more liquid nitrogen.
There is liquid
It will
nitrogen.
happen. Excuse me.
So once you
Where's the
see that
liquid
it's
nitrogen
not
can?
like getting powder or anything, in middle you keep on adding little liquid nitrogen.
And when the liquid nitrogen is completely dry, then only
Also, you
pour
can
fish.
separate, but the contamination chances are more while using this. Because RNA
Pour
will be
only a
more
little bit
DNA will be
and
somewhat
slowly so that it
immediate.
doesn't bubble out with the
So the
nitrogen.
percentage of
Slowly.
contamination chances are more while taking DNA.
No.
Maybe you can treat
If you put like
RNAs
this, the bubble will
after
go in the
isolating the
flask.
DNA.
I'll show.
If you treat
Just
RNAs,
take like
RNA will
this,
be
slowly.
DNA will be intact.
Don't be so soft.
Done.
Grind it harshly. Yeah.
But this
Yes.
method,
If you
Good.
keep it like this, then also the bubble will come
It
inside
has
the
to lose
flask.
all its green color. It has to change into an ashy green.
And when you're adding, immediately you try to collect everything. Yes, first collect everything from the walls and all, and then start grinding it with more pressure.
Now you guys are doing it?
Yes.
Nice. Once it completely dries out, once the fumes also stop coming, then you can pour some fresh. And
It's
when you're
okay.
pouring, be careful. Slowly.
so proteins high molecular weight. Yeah, targets some other.
Done, no?
Guys, everything is fine. Going good.
Or pots.
Give that.
You should make it fine powder.
लेकिन ना doing that उनको चाहिए
Completely over. Just ask. They will be
We
giving
don't
you
have
the liquid nitrogen.
flask.
Fine powder. Like
Over?
very much fine.
Okay, then I'll add in
ash
yours.
green time पूछ रहे हो color पूछ रहे हो
Oh, wait.
color ash green
See, now it's coming in
it
powder.
should use
Now,
most of the green
you need liquid nitrogen?
हाँ
So,
जब
let's
पूरा
take
dry
this.
when it gets all dry
Others can also try, not only one person.
Yeah.
Give it to other
इधर दे दो
person.
थोड़ा इनको slowly डालना it will bubble up slowly
What to do? Yeah.
Hold it in the top and press it and grind. Hold it in the top.
Can't
Yeah.
see. What is he doing?
Move your hand.
Very harshly you have to do. You are mechanically breaking open the cells. So that should be the
Show on. Did you pour? You are
इनका
mostly
नहीं
done.
जलेगा.
You are mostly done. Maybe just. Do you want me to pour some liquid nitrogen?
इनका नहीं जलेगा हाथ
You
में.
want to try? I think you have to. Come. Slowly first with the then total solution. Otherwise, I will.
श्रेया, maybe
I
you
think
want
this
to
is
wear
your last
gloves.
go.
Wear gloves.
Do you guys need some liquid nitrogen? Show.
More pressure,
Fine.
little
I'll
bit.
pour. You guys are also almost done. Keep it down.
Yeah, group 2. How about you? Is it fine? Grinding is going on? Be very
Sometimes
harsh.
when we grind,
Fast and
like
Okay.
for example,
Now
in my case,
grind
I
it
grind for
very
rice and
quickly.
all.
Jaldi.
It becomes very jelly. So what
Jelly,
what
jelly. So
One after
liquid
the other,
nitrogen
let's go
more you
for
can
missionaries. Everyone
put.
cannot go.
more?
Yes, little more. And also maybe you can try when you take in Falcon, put some liquid nitrogen directly. Suppose if you are grinding climate, so you see it is immediately coming out of them. They add little liquid nitrogen, not much, and then keep on grinding and pressing it. So once the liquid nitrogen goes, it will start getting dry, just like lyophilization.
I think we can just directly take.
Directly will take.
We directly will take. Not an issue. Later we have to add 1 ml Trizol, right?
100 milligrams of
I was thinking we can give each one, and they can do individually. One, up and off. They all can collect for each of them. Because it's like
No, don't take everything. Take half. Half take.
this one person is doing and rest all are just. Can we get the liquid nitrogen? Wait. Liquid nitrogen?
इधर can इधर इधर खत्म
Where
हो
is
गया
the
उसमें.
can? Okay.
Show once.
Can देना?
Can नीचे ही रखा है नीचे ही है. Okay, you guys are almost done. That's ashy green. Can I see your color? Little more. Can I see the color? थोड़ा सा और. I'll get liquid.
So next step you will be transferring.
इन दोनों ग्रुप को चाहिए.
From now onwards,
इसका हो
every
गया.
process will be done in the epilogue groups. So you can follow that. So you'll be adding a trizol followed by whatever the steps they mentioned.
Wait. Now grind it very quickly.
Not that.
Excuse me. I'll put some nitrogen.
There will be a small epilogue we'll be giving. And avoid speeding much while RNA isolation. That is also very
This is very
It
soft.
also causes
Be
contamination.
harsher.
For now it's okay. But when you do for your actual experiments, speak less.
You can see how it's changing color, right? I'll put some more. Why aren't the boys doing it? It has to be very harsh.
So one after the other
Wait,
group,
give
let
me
them
a second.
start.
I'll put some more. And then with
Anyway,
all
they're
your strength,
not going
just
to win. No?
it's almost there. You guys are also almost. इनका हो गया है.
I think
नम्रता ग्रुप 1 का हो
almost
गया है.
done.
ग्रुप 1 का हो गया है. इनका भी हो ही गया है. Shall I
this
put
much
some more?
whitish green is the
I think it's
endpoint. You can
it's
decide.
fine.
But you
Yeah,
you
it's fine.
No, add the liquid nitrogen, let them come at one together.
see
So the
now if
process
you see
starts.
little bit moisture you can see. That correct? So you should not take it. Now see the
Yeah,
lot
yeah,
of
it's
moisture
here.
is there. When you
It's
take,
here.
you can't. The RNA will not come proper. So once you see this, you immediately add liquid nitrogen, and then you should not give space once you collect the pellet because the moisture will come. So now you can see this is like this. Maybe add little bit of liquid nitrogen and then again.
Powder it and transfer.
Yes, entirely powder. It should not be
कहाँ गया
what?
इनका
Yes.
sample? यहाँ इनका sample कहाँ है?
Hey, see me.
Put on the this plates.
I think you guys need to scrape it off now with the spatula.
I'll show.
Show your color.
Shreya, liquid nitrogen. I'll show it.
Yeah.
So
This
this
is fine.
is the one.
This is
First
ash
you
green.
will
This is
pour
fine. Now
liquid
I'll do
nitrogen with the
epilogues.
motor
You put the entire
Okay.
powder.
Then immediately when you put, first you collect everything from sides. Okay.
Yeah, these both
Once
groups are
you
done.
collect
I'll
it,
give them 2 ml epilogues.
now you see it's getting dried
That
now.
is 2 ml, yes.
Keep on collecting the all parts. Now it is collected.
And gloves
Now
always
you keep on slowly
make sure
grinding.
your
Once
gloves
this is gone,
does not
now
have
with lot of pressure you have to grind.
transfer gently
You can see.
into the tube.
They come.
Yeah, you can start.
Now take the Eppendorf and scrap it
This is the Eppendorf. Collect all the powder here. Okay.
from that, and immediately you should take. The more faster, others moisture will come.
Full powder.
Generally we take
After
130, 140
transferring the
mg.
powder, please add the trisol
No,
immediately.
you don't have to take full powder. You can take aliquots, 120, 130 mg.
So after taking, you have to weight it. Na?
Yes, sir, weight.
Weighing.
Yes, 130 mg. But now,
Along with the tube.
yes, along with tube.
After putting the
130 mg you
powder,
should take,
remove the
130 or
trisol, it's
140,
in the bottle. 1
depending.
ml of trisol.
Then it will come 100 mg, I think.
1 ml.
No, no,
And
120
keep the
mg it will
protocol
come. But
like 1
generally we
ml
prefer to
have
take
the
140 mg, 20 mg more.
So 130 mg sample will be, we have to take it.
Yes, yes, sir. You will
Show
keep
one.
the Eppendorf, tear, and then you will put 130 mg immediately, and then yes, yes,
Show me how.
and then you will collect different. Maybe collect aliquots 2, 3, so that you
Put
can get
1
better RNA.
spoon more and then quickly add the trisol. 1 ml trisol.
Which step
The
you are
pipettes
in?
and tip boxes are here. You guys know how to handle it.
Immediately, moisture is coming fast.
1 ml, 1 ml, the green one. Yeah. Yes,
Add.
yes.
Maybe you can make more on Eppendorf more. Yeah. This also
This
you
is
can
not 1
scrap.
ml.
Yes, this is trisol.
Like it's turning into liquid while adding.
It's a Eppendorf was contaminated with water.
I don't know, like when we are touching it.
Actually, you don't have to give time. You don't have to touch this. You don't have to immediately pour inside the Eppendorf. Now we'll do one thing. Pour little bit liquid nitrogen in this, and then immediately now see this moisture is there. So also pre-dip this in also liquid nitrogen because moisture can also be here. I'll show maybe once.
Where's the protocol book? Does
Liquid
it
nitrogen.
say to add chloroform immediately? Add 200. Add 200
Little bit
microliters of
space I
chloroform
can
as
get.
well. This is chloroform. There's a 200 microliter
So once you
pipette.
add, I'll show. Once you
Is
add,
there?
first you
This
collect
is
the
100. Add
cells
2 times.
from the walls.
Okay.
You can see the cells are there.
This is the tip box.
And once it is less, now you start grinding.
Give the chloroform.
It's not grinded properly. Did you wipe that? Okay. Now you dip this in liquid nitrogen, and then you have Eppendorf. This different one.
Okay.
It will
Done.
cause
Now
again
invert it
moisture.
gently. This is. You guys added 1 ml of trisol.
1 ml trisol is added.
Perfect.
Now chloroform 200.
This is 200 only. Just add once, and then you have to invert it gently. Chloroform. It's the.
Maybe you can. I just showed you. You
This
can
is
also collect and try on your own. It has lot of cells. Now you try and add more on your own. Put liquid nitrogen. Now you see the moment moisture comes, you put little bit of liquid nitrogen so that
And
it's in fine
also
powder, and
put the
then dip
timer
this in
to 3
liquid
minutes. We
nitrogen
have
so
to
that the
incubate
moisture
it at
doesn't
room
come from
temperature
this.
for 3 minutes.
Now try, I'll see.
Follow those 3 minutes means exactly 3 minutes.
Little, yeah, not much.
Just once you have.
Mobile, you keep the timer.
And collect the cells, grind it, and immediately take.
You just follow your.
Okay, now you have to mix it gently and put a timer for 3 minutes on your phones. 3 minutes time. You easily grind कर रहे हो.
उनका सेल्स नहीं था ना पूरा वो पानी हो गया था देन दे आर अगेन कलेक्टिंग लिटिल बिट
ठीक है अभी ट्राइजल मैं तो बोल रही हूँ
ये सब लाइव चल रहा है Done? Yes. Now you understood?
Now I feel what actresses feel. बोलो मत PR है ये
Yeah. Done? Which step you guys are
Show
in?
once.
We added chloroform 200 microliter.
Fine,
and
fine. Add 1 ml of trisol.
2-3 minutes. Okay.
ठीक है
after that centrifuge.
Then I'll keep on room RT 4 degrees.
Incubate at room temperature.
No. A centrifuge you have to do at 4 degrees Celsius. Did you keep?
नहीं no. RT लिखा हुआ है इस पे भी इसमें 4 है ना Okay, fine.
Then after chloroform, room temperature.
Room
Protocol
temperature
it's
only.
written.
Put a timer for 3.
Protocol it is written, see?
Huh. Keep it in room temperature in the ice. Room temperature 3 minutes. You have keep the timer for 3 minutes. Then we'll centrifuge it at
4 degrees.
4 degrees. Guys, are the trisol? This is not the pipette. This is the one. This is the tip.
डे वन का फोटो नहीं लिया था क्या दुश्मनी थी
क्या?
मेरे से एक फोटो नहीं लिया डे वन का है दिखा देना प्लीज
Fine, fine, fine. Take it out. Take it out. Beat. Chalo, chalo. Okay, now add 200 microliter of chloroform.
If you are delaying, the other groups will suffer. Everyone?
Do you have? If you have 100, then you can add 2 times. This is a 200, yeah. Add once and mix it.
now once this work is done keep it here always clean the place which you are using
हो जाएगा ना इनका भी शुरू होगा 3 मिनट बाकी इन दोनों का होगा
okay they added chloroform
they added chloroform
now they have to centrifuge I kept for 4 degree
Guys, be a little faster. Because every group need to.
You guys are almost. Your 3 minutes are done. 3 minutes are also done. Keep it there. We'll put it for centrifugation all at once.
So group 1 is done. What about group 2 and group 3?
group 2 done
Yeah, once you are done with the incubation, let us know.
mix it gently
Gently.
gently
Inverted. Yes.
like this
Like
yes
And now put a timer for 3 minutes.
How is this done? 3 minutes.
3 minutes are done. Okay, we are just waiting 2 minutes for them and then we'll do it. And tab tak you just clean your workbench like these things. It has to be tidy at every point. Fine. Tab tak you guys clean the workbench also because RNA has to be done in very tidy conditions.
it's okay
Fine, we'll take it. We'll collect it from you. We are waiting for one group for the 3 minutes. Then we'll put everything on.
So now after this, after this you will have 3 different phases I told. So you have to collect the upper phase very properly so that you should not collect the middle phase. Otherwise contamination will come. After that
If you want more, then you
you'll add 40 microliter of NaCl which will help in precipitation and neutralizing the charge and then 500 microliter of isopropanol. And after that we'll see. If there is any confusion, any step or anything you want to know, ask.
Yeah, accordingly you can increase. Anyway, you will break open the cells. So the final yield will vary depends upon the how much initial cells you are taking.
Go through the protocol.
So if you take less cells,
steps
the
they have.
concentration
Where
will
do you
obviously
have
go
to
low.
save the supernatant?
If you
Where
take
do you
more,
have to discard the
logically
supernatant? That is why
your yield should be very high.
people often confuse.
But you don't need very high concentration also.
So you should know when you are saving the supernatant and when you are
ctNA
discarding.
conversion.
Yeah, of course.
That's enough.
Sure.
The quality is matters. Quantity is
Done layer incubation.
इनका बस 2 मिनट बाकी है बाकी सब
If
का
your
हो
quantity
गया.
is something 5,000
them forward.
nanogram, but your 260 by 280 is gone. No use. I can't use anything else. It will be done at the time of tea break only, not 3:30, 3:30. Exactly. You want? But you have to give.
But this is cute, huh?
So after putting the centrifugation, I will be giving intro for the next experiment so that because 30 minutes is long. Simultaneously we will.
वो देख रही हूँ रूम 30 हुआ 4 डिग्री.
Result will take almost 2.5
दीदी उसको बहुत बोलना है.
hours. So you should be patient enough.
Sir, I think now we can take one.
Yeah, I'm done. That's it.
No, no. Now you will tell the colony and everything.
Yeah, while
no?
starting
हाँ.
next. Fine.
We'll put for centrifugation. I think we should put.
Yeah,
It's
okay.
21.
The team 2 is done.
10 मिनट और लगा देंगे. 40 minutes.
बोलो.
3 minutes is over.
Okay. You can start.
Yeah, yes.
You can start.
Give me your tube.
Yeah, yeah. Everyone mark your group number. At the end, you will not identify who is which one. So either every group should get the quantity or none of the group should get. Okay. So mark it, but keep in ice. Keep in ice. Keep in ice.
Markers here.
Marker.
Label your this with groups.
You
Group
can label
1,
it
group 2, group 3.
No,
Okay?
no. Group 1, 2, 3. They already know. 1, 2, 3. Let it go.
This was group 1.
This is group 1, 2, 3.
This is group 1, sir.
So, okay, there is a clash
1,
for
2,
group 1.
3. Yes.
Fine. Anyway. Okay, you decided you are group 1. Okay. Let's see the final yield.
Yes. 3, 3, 3.
No, from once you break open the
1, 2,
cells,
3.
it's
1, 2, 3.
always ideal to keep it in the ice.
3. I'm telling 3.
You keep it in the ice. So even centrifugation has to be done in 4 degrees Celsius. Okay. So RNA is very sensitive to temperature. Yeah.
Achha, what you will understand when you see,
why they are
you
kept
should label it
Joel
properly.
small
See, once you
letters.
label, you should label and you keep it. So then sometimes
that's-
it gets removed.
I
So
don't.
you
That's
can't
just a
understand what is
company
which is
name,
your
the
sample.
commercial.
Joel is company name.
One,
No,
उनका दे दो.
trizol. That's it. Solution zol.
Like these are the basic things which
There is no
you will
specific
understand
meaning
when
to
you
my
start
knowledge.
doing.
It's a monophasic
Very
solution
normal
of
things
two
you
things:
think, oh, this is, but you will
guanidine,
face a lot of problems.
isothiocyanate,
I face.
as well as phenol.
So that's
These
all
two
I'm
things are
telling.
added,
Experience.
and they make it as a single
You
solution.
guys are
Earlier, it
in
was separately.
graduation.
They need to add
Masters.
guanidine as well as the phenol.
You both.
So they made it as a
She
single
also.
solution, commercially available. That's it. The history of RNA isolation using this phenol method starts in 1970. At least 1966, I think so. Yeah, where they used initially everything will be separate components. Phenol will be separate.
I'm putting for 30
Yeah.
minutes.
No, no, it doesn't
So
work
phenol will be
like that.
separate,
If you don't
chloroform will
get
be separate,
experience, they can know
guanidine.
what they
Guanidine
don't
first time they
know.
didn't use it.
You can start with what you
So
know.
what is the problem if you didn't use guanidine? Guanidium isothiocyanate. You know trizol has two components, right? She explained it. One is phenol, other one is guanidium isothiocyanate. What is the role of guanidium isothiocyanate? What is that? Yeah, partially you are correct. What is the biomolecular? No, no.
साइंटिफिक रीजन था कि ट्राइजल बहुत बड़ा हो रहा था.
Okay,
I just
I will
compressed
see
it.
what are the answer. Pass it one after the other. Say what's your
No.
answer.
Guanine.
Guanine. Okay, she's telling guanine. What about you? No, your answer also.
I don't know. Maybe protein or.
So, question is clear, right? Guanidium isothiocyanate is used for what purpose? It will denature a particular biomolecular, as she said. What's your name? What's your name? She just said partially correct that it will denature a particular biomolecular. What is a biomolecular is the question. Yeah, perfect. What's your name? Yeah, Tanuja said it correctly. So it is mainly used to inactivate the RNase. If RNase is there, it's going to degrade the RNA, right? So that's why they introduced guanidium isothiocyanate. So from then onwards, they made the common solution trizol, which has both phenol for denaturing the protein and guanidium for inactivating the RNase. So that's the history stuff. So I think now please put the incubation centrifugation for
I थोड़ा edit कर देते तो बेस्ट है.
So instead of waiting for that, we will have a substance about our next experiment. What has to be done.
नहीं ऐसा है आईफोन में you can edit him also. हाँ.
So now we are not going to do the cDNA synthesis,
So
but
है editing try करो.
anyway, you should be knowing if you are going for any RT-PCR, you will be doing with the cDNA. What is the difference between normal DNA and cDNA?
Sticker you can make and then send it in GIAN group.
Yeah,
Sticker
everyone
you
says
make.
compliment. ATP,
हाँ, इसको करेंगे.
mitochondria is the powerhouse of
अब
the cell,
इसको
something
बात
like
करेंगे
that.
WhatsApp पे भेजेंगे किसी को.
So explain more about
हाँ.
what is cDNA.
और GIAN group खोलेंगे.
Okay, so RNA is generally an unstable compound biomolecule, so it will degrade very fast. So we have to
नंबर तक भेज दीजिए.
take that in. I mean, the double stranded is always a standard. So we have to transcript the other like nucleotides using like primers and all.
Yeah, you're almost correct. Perfect. But what is the difference between the actual difference between the normal DNA and cDNA? Is there any difference or it's just a DNA? Or both are similar?
Reverse transcript is way of RNA which we use. Like
CD
it's just the opposite
complementary pair of RNA which we use.
Okay, so what will be the difference between the actual genomic DNA versus cDNA? What might be the difference?
It's single strand.
No, not single strand.
Normal DNA is longer as compared to complementary DNA. We can say as some basis.
Longer, you mean to say?
Normal DNA is longer as compared to.
What is the reason? Yeah, you are almost there.
Because introns are because after post transcriptional modification we make complementary DNA from mRNA.
Yeah.
That's
So
why
splicing introns will be
splicing
removed.
intron, yeah.
That's
Removal of
actually
intron.
you will be having only the open reading frame. The actual answer in cDNA you won't find any introgenic region. So only the stitched ORF. So complete genomic sequences only for coding for the gene will be there. There won't be any introns. That's the advantage of using cDNA. So why do we use cDNA for RT-PCR instead of using the direct genomic DNA? We could have used genomic DNA also, no?
It's a huge, genomic DNA is very huge, but
Yeah.
the gene of interest is very small.
Yeah, it's like wasting your resource, first of all. If you want to just check whether the expression is there or not, we can directly, like for example, your cDNA or a particular transcript for a gene might be 500 bases. But with the intronic sequence, it might be some 5,000 bases, 6,000 bases, it varies. So that's the advantage. We are using cDNA to minimize the resources wastage as well as the time. The amplification time will be reduced. So that's the major reason we are using for cDNA. So basically the cDNA synthesis will be following the same protocol. Shailaja is here. Shailaja, just change the slides to. No, not here, not here, not there. So you will be using the components required for the cDNA synthesis. As usual, you know RNA should be there. Apart from RNA, you will be using oligo-DT primers, DNTP, and RNase-free water. So I think you might be having what is oligo-DT primers? It's a primer, first of all. So RNA will have poly A tails, right?
Yeah.
So to capture that, they will be giving you the oligo-DT primers. So they will go to bind them, and any transcripts will be amplified. All the transcripts. There may be thousands. At that time point, there may be thousands genes has been transcribed. So all those transcripts will be converted to the cDNA form. So this will be done in the PCR machines itself. So now we are not going to do, but I am just telling you as a glimpse. So if you are going for any RT-PCR, the RNA isolation, the whatever RNA you are isolated after confirming the quality. So as I was discussing with him, quality important or quantity important? What will be your answer? So quality is very important, right? Even if you get some 100, 200 nanogram, is fine. But your quality 260 by 280 should be around 2. So otherwise you cannot use for any experiment. So that way, so once you confirmed your quality is fine, then you should go for the cDNA synthesis. So after cDNA synthesis, so we will be doing the PCR. So this PCR, we will be starting this PCR now. But only thing, we will be using the genomic template. Instead of the cDNA, we will be using genomic template from a single colony. So individual colony, we will just pick a small colony using a toothpick, and you will be putting inside the mixture, PCR master mixture. So any of you say what are all the master mixture components? Can show. Polymerase.
Tag polymerase.
Yeah, any polymerase. Tag is a old. Now there are many polymerase. Polymerase it should be have DNA polymerase it should have. Apart from that, DNTPs. Yeah, primers are very important. Forward primer, reverse primer. Yeah, RNase free water. So these are the basic components of any PCR. So how many of you have done PCR earlier? You guys not done. So okay, today you are going to do. Don't worry. So for you, I think I will be giving you explanation. At least theoretically, you have some idea about how the PCR works, right? So that you will be having, right? So the double stranded DNA you will be giving as a template. So the primer, the forward and reverse will go and bind to a particular target region, and they will be amplifying. So there will be two steps: denaturation, annealing, and elongation. So denaturation, what is mean by denaturation?
It's double stranded DNA to single stranded form.
Yeah. So why it has to be done? Yeah, perfect. It has to open up so that the primer need access, right? If it is double stranded, they will be bonded by the hydrogen bonds. So in order to get the access, the primer, the temp to double stranded has to be open up. So we are heating at 95 degrees Celsius, followed by annealing. So this annealing, how will you identify or how will you fix for your primers? Is it any particular annealing temperature is there for every primers?
I think at 42 degree, I don't know, 40, 60, 60.
Yeah. What is that? You are almost there. Yeah, TM, melting temperature.
Melting temperature.
So yeah, melting temperature. Yeah, every primer has a particular melting temperature. It depends upon how many
Thiamine or
ATGC.
GCP
So GC has more melting
more
temperature
melting
or
temperature,
AT?
GC more melting temperature, AT less melting.
Yeah, because the bondings are more stronger in the GC
GC,
content, right? So if your primer has more GC content, then obviously the TM will go very high. So accordingly, while designing the primer, you need to know or you need to fix in a way that it is ideal. The primer annealing temperature is ideal. So then elongation is common. The polymerase is going to just add the nucleotides, and maximum you can keep 30 to 35 cycles. So at the end of the 35 cycles, you will be having only your target region will be amplified from the entire genomic region.
Why DNA polymerase cannot start the synthesis of a DNA strand?
Yeah, that is a good question actually. Do you know there is a difference between RNA polymerase and DNA polymerase? So it's by nature itself the DNA doesn't have the in vivo, or I can say the exact term is it cannot be able to initiate the synthesis without any primers. Whereas transcription, the RNA polymerase, they doesn't require any primers. So by nature they have been designed in a way that they require some primer.
RNA polymerase is more complex as compared to DNA polymerase. Can you say that?
It's not complexity, it's the nature itself. They like the proof, for example, DNA has a proof reading 3 prime to 5 prime. So in nature they have been designed in a way to have its own efficiency. We are exploiting that to make our own benefits. For example, if they don't require the primers, in that case accordingly if DNA able to synthesize by itself, then we can PCR will not be into effective, right? So science will be born by just thinking about how it works.
My question is like RNA polymerase is made up of 5 different kinds of subunits. Like that, what is the structural composition of DNA polymerase?
No, it's like one will be catalytic, another will be regulatory subunits. But exactly they couldn't identify why the reason, exact reason there is no. They will be telling that they cannot. But exactly still there is no answer for it, I think so. So but RNA polymerase for sure it can do, that we know. Transcription they can start any point without any primers, they can do it. So that's how the nature works. So we are just trying to exploit the natural phenomena. Like similarly the cDNA also. The enzyme where they reverse transcriptase, where do you think they have isolated? Which organism? Retrovirus. So retrovirus has the ability to convert RNA to DNA. So we just observed that and we are just implementing that for this. So all our experiment will be somewhere inspired by some natural phenomena. So we are trying to exploit it for our requirements. So that's how science progresses. So now we are going to do the same PCR, but from the colony. So I will be giving you a plate now. So we have to make a mixture first, master mixture. I think I have been given you the protocol, right? In the protocol, I have just opened the protocol. So in that PCR master mixture, I have mentioned you. So I have mentioned how to make the master mix. I will be giving you the whatever the required enzymes, everything. So accordingly, you will be adding to the PCR tubes. So group wise we will be giving you. You add accordingly, and finally the template part we will be giving you a single colony. We will pick a single colony. I will show you how to do. Pick a single colony and drop it inside.
Do we need to lyse it prior or?
Yeah. No. In the first we need to lyse it. I will be telling you. There will be 2 steps I would have mentioned. The colony has to be lysed in the Kellex 100. There is a disodium polymer. It's actually a polymer that's help in purifying the debris from the genetic material. So we need to pick a single colony, put it in the 50 ml of 50 microliter of Kellex 100, and heat it at 99 degrees Celsius for 10 minutes. And after that, we'll just centrifuge short spin we'll be giving, and supernatant we will be adding 1 microliter to as a template to the PCR mixture. Any doubt here you can ask. Yeah. Yeah, I discussed the point number, the colony PCR column I mentioned, no? In the protocol, there is a heading called colony PCR. Can you see that? So in that I have mentioned how to isolate the genomic DNA from the single colony. So how many minutes more for the centrifuge? 20 minutes more. Okay. Okay, fine. 20 minutes more. Okay, then I think, yeah. Colony isolation we can do. Fine. Yeah. This is the same Chlamydomonas colonies. But any colony you can try. The method will be the same. If you are using E. coli, it will be more easier. You don't need Kilex for E. coli. We can directly drop in the water and heat it in 99 degrees Celsius, 100 degrees Celsius for 10 minutes. Centrifuge short spin and then take the 1 microliter. Even I heard E. coli, you can directly add to the PCR mixture also. No need to lyse the cell separately because their cell walls are very fragile. So in the PCR protocol itself, they will get break open. In higher pants, there is no colony PCR. You need to use CTAB method to isolate the DNA. Whereas CTAB method, CTAB they used. It's like a trisol only, well standardized method. CTAB they used to say CTAB. Cetyl methyl. Yeah. So I think there are 20 more minutes for the process to complete for your RNA. I think we can start for the single colony genetic material isolation first. So meanwhile, it will be done. Okay. And maybe after the steps around 3:30 or 4, you can go for tea break. We'll be a little faster so that we can complete by 6:30 at least. What time you want to complete? It's okay. No, her reaction was too much. I can feel like she was very much surprised. What time you leave by every day? 5:30. That's why I said today it will be little more. It's not the same like biophysical. Okay, I will bring the colony. Just I'll be just showing you one. You guys will be doing the remaining part. For the culture, safety is not possible to even for the start has to be done in the lab. This is a okay, I'm just taking 50 microliter of the flex plant. Whenever you take it, try to mix it and take the slurry. Along with the slurry should be there. Don't take the super. That's it. Slurry will be there. Now I'll show you. Give me a marker. Yeah. Sorry. Yeah, marker. I need a marker. Yeah. I'm putting my initial. You put your names. So okay, I'm going to take, and everything should be autoclave. Take a single toothpick, and everything has to be done in the laminar. Okay. For now, it's fine. I'm taking a single colony. Single or small portion is fine, as less as possible. Okay. Just spill it like this so that everything will go inside. Okay. That same way you guys also do. Take 50 microliter of the polymer, Kilex 100. Label group 1, group 2, group 3. Okay. And then add it and take a single colony from yours. You can try one can take this. This is the toothpick. Just take 4 tubes first. Yeah. Close the bottle. Yeah. Use a toothpick and you first add the first add 50. Mix it and take 50. Yeah, add it. 50. Right? depends upon, that's what I'm telling. She's asking a good question. I think I explained, maybe she would have missed it. She's asking directly, why don't we put it in the PCR mixture and go for the PCR cycle? Why you want to add additional step for lysis? So why do you require that? That's your question, right? Why you want additional step? The question is simple. She's asking, we are already going for a PCR. We can directly add this colony to the PCR master mix. Why you want to do separate lysis steps? She's asking that. Okay.
Anyway, we are providing higher temperature here, so it will break.
Why?
Anyway, we are providing higher temperature for denaturation, so automatically it will break.
It should break, she's telling. 100 degrees something we are 99 or 98, 95 to 98, 100 you will be giving for the initial denaturation. She's thinking, why don't it break? The question is depends upon the organism you are going to use.
Because like
The chlamydomonas is very rigid. Initially, I also tried to do with the chlamydomonas. The cells are very hard. So it requires either mechanical pressure, like for RNA we did it, or like separate heat inactivation separately, and you need to spin down with the Kelex 100 so that debris will settle. If you directly, bacterial no need. For bacteria, we can directly take a colony and we'll put it in the master mix. That's enough because their cell walls are very fragile. That's what I said. For this, when I tried, it doesn't work. Then when I gone through the paper, they suggested to use this technique. It has the walls are very hard. Yeah.
then just temperature is not enough.
Yeah, not enough.
This reagent is necessary.
Yeah, this is necessary, really necessary. There are different methods. Some people will use even 70% ethanol. So everyone has their own optimization. So I found this is more helpful for me. So I started implementing this.
So Kellex 100 is made up of
It's a polymer disodium salt. It's a ready-madely available in Biorad available. It's like a big particle with a mesh size. There will be different mesh. So what they are trying to say, what is the advantage of using this is, once you break open the cells, they will go bind to this material and they'll pull down. After we lyse this, we'll centrifuge. So debris will settle. Only the nuclear material will be at the top. So while taking, we'll get only the fresh or pure template rather than the contaminant.
Another one thing, we are taking her chlamydomonas, and the cell wall of chlamydomonas is generally made up of cellulose. So, in which step cellulose is broken down?
Sorry?
In which step cellulose is broken down?
In this, or you mean to say?
In this, we have to perform PCR. So for PCR, we have to isolate DNA first.
First, we need to isolate DNA. Right now we are lysing the cell. After adding this after adding this polymer, we will go for lysing 10 minutes at 98 degrees Celsius or 100 degrees Celsius for 10 minutes. So that time the cell wall will be broken up. Then we need to spin it, short spin. Then we'll take that template for your PCR experiment. Right? Got it. Right now I'm using one autophagy gene, ATG. So any gene you can take it up. So since I works with ATG, I just took them as an example for you. So it's autophagy, you know, right? Autophagy, catabolic pathway, cell death. They works in that. So any gene works the same. If you design a primer properly, I will be telling about how to design the primers in shortly. Maybe when we have a time, we have still time, lot of time. We'll discuss about it. It's over. So how many groups done? Only one group. This is done. This is mine. This you are starting. Okay. Okay. Nothing is there. Shreya, they need Kilex. Add some more.
They need what?
Kilex, the tube. No? What happened? Okay, they didn't have anything. Yeah. This tube itself we can add nothing now.
We have already added this culture.
Where? Here.
Give me the small.
How?
Give me the. Yeah. Give me that. Just add. Yeah, 200. Okay, she would have not noticed. Maybe. No problem. If it is colony is there, nothing will happen. Ideally, you should have dropped some colonies into it. That's the logic. If colonies are not there, you won't get any template. Take 50 and add to it. If you are not sure, then you take more. Slow. While you are in a hurry to remove the pipette. Wait. Give me. I'll show you. So pipette handling is also a skill. So in the pipette there will be two kind of pressings will be there. First will be light, further will be the hard press. So while sucking the media, you should always give only one press, single press. You should not go for the double press. Okay. So single press, take it. I think there is air bubbles. Slowly you should remove it. The leaving should be very slow, otherwise air bubbles will come. Then only it will be complete. And while dropping, you should give a double press. I will show you double press. Double press. Okay. So gently you release. Single press to suck it. Slow. Slow. You are again very fast. Yeah. Okay.
Is it okay?
Fine, fine.
So I have to add.
Whatever tube yours, you can give me this. That's fine. You labeled yours, right? You drop your colony. If you are not sure, take some more colony and drop it. Who is the other group need to be done? You are. I can't hear properly.
I also want to try, sir. I don't.
Yeah, fine, no problem. You can do it.
I don't have tube.
It's nothing. Just it's even one tube is enough. You are just going to take a single colony and you're going to. You can inoculate in the same tube. Nothing will happen. Just going to drop. Just will it spin the toothpick. Can you tell them what is that? Oh, it's Kelex and Kelex. It's just a polymer. It's available in the ready-made form, which is disodium. It has a different pore size mesh. Based on your biology chemistry. No. This is for any algal cells. The main purpose is they will remove the debris. They will separate the nuclear material. DNA will be in the aqueous portion. This will pull down the debris down.
What is the 50 micro?
So that's the control of. One more group required? Yeah. Maybe you add, let her inoculate. She's feeling she want to inoculate her yours first. Use this for Kelex. You are looking for this. You add the thing, let her inoculate one colony. Pick a colony. She very. Yeah, 50 already. Yeah. Be very careful. Don't be too harsh. Pipette will also go. So single press, take it. While releasing, slowly release. Okay. No. While removing, slowly release. Don't need. You are pressing double press. You are giving double press. No. You are completely removed. Yeah. Now double press. Only single press. No. You are giving this much. No. I'm telling this much only. And you take it inside. Take it inside and release. See my hands. How I'm releasing slowly. Slowly. You taken more. If you are pressing double press now, it will take more volume than the required. So always single press. Release slowly. Yeah, take it inside. Release slowly, slowly. Yeah, slowly. Fine. That's a perfect. Add it to the wall. Fine. Done. Let her inoculate. It's like a. There is nothing. Use this. Take a single little quantity. Use this tip, toothpick. Scrap a small portion. No, let her do it. Be faster. Don't open it for long. Yeah, you can see something, right? Colonies. Then you inoculate. Mix it. No. Just do this. Slow. It's done. Hold on. Fine. No. Let be a blending. Done. What happened? Any problem?
Only do one more colony.
You can put it in the same. It's nothing. Use different toothpick. Take an. Nothing. This is very simple. Nothing complicated. For entire reaction, we require one tube is enough because you will be having at least 30 microliter, 50 microliter you are adding, right? The debris will be something around 5 to 10 microliter. So you will be definitely going to have 30 to 40 microliter. So per reaction, 1 microliter. That's more than enough. Just I want to make you give a hands-on experience. I am making you 3 to 4. This is colony physics. Then RNA we are doing RT-PCR. Yeah, that is for RT-PCR, but we are not doing it now. We are only isolating RNA. Just a demonstration of RNA isolation. For entire program, that will take long time. CDNA also? No, CDNA. I'm just showing what is how it has to be done. Done? So be careful. Take it. Yeah. Okay, all the 4 groups done, right? One group is missing. Okay, it's including mine. 3. Okay. Okay, this is fine. Minus 3. This is fine. Fine. That's now what we have to do? We'll just heat it at temperature I mentioned. 98 at 12 minutes. 10 to 12 minutes is there is nothing like perfect. And so I'll be showing you come. Or you can come group by group. It will be probably right. 5 to 6. It's remaining people you can stay there.
Okay. Yes.
ओके तीसरा ग्रुप क्या है अभी वो फर्स्ट गए हैं जेरोम के साथ वो बस एक बात
ये ऊपर वाला लेना है which is group 2. This is group 3. Yes. Now you can see different differences. This is upper interface and this is proteins and lipids, last bottom phase. This is your RNA. So when you're taking, take so that this should not touch interface. If it touches, then DNA will come along with RNA. The upper layer is RNA. This is group 2. I'll give you a fresh setup. Just collect the supernatant very carefully. You guys know how to use a calculator. कोई दिक्कत हो just call us. थोड़ा सा बता दोगे एक बार
centrifuge RNA is come.
Yeah, your group was busy there, na? Sure.
This one only.
This is 200. Then yellow. Fast. Take it a little faster and then we'll start stabilizing.
The transfer.
Now till now you can see. Till little bit more. Now don't take it. Yes. Now what is the next step? Where is the this protocol? Now. Now you have to add 40 microliter of 5 molar NaCl we have provided you, and then add 5 microliter of isopropanol, and then mix well gently, and then keep it incubate at room temperature for 10 minutes. Okay, then we'll centrifuge again. So also when you're taking, take it faster. Like don't keep it. It doesn't depend on which pipette you're using. Give. Anything. It's okay. Give. Let them take. No. Can we use 100 or should we use 200? Should we use 100 or should anything? Just to collect. There's no rule that you have to use some pipette. We can directly take 1 ml also. Yes. Just have to see that the interface should not come along with RNA. See, if you take this slow, if it touches the interface, you can see the interface. Wait, you see the interface is there. This is DNA, and the bottom is proteins and lipids. So it should not touch the interface. The white is DNA and the green is proteins and lipids. The entire transparent thing we have to take off. Yes. So the more you can take, yeah, that's what you can take fast. Otherwise, it starts any pipette and then little bit tilted when it comes to white. Now you will touch. Now you take like this and then you take. Don't touch the interface. Done? Now you have to add 40 microliter of NaCl and isopropanol and incubate for 10 minutes. 40 microliter of NaCl. No. See, you took. You didn't take. Take it fast, otherwise it will start with the. Oh, any tube you can. See, any pipette you can touch. Take this pipette. Take slowly. Any pipette you can. You just have to take the transparent part. Give that. Now take. Take it slowly. Single press. Wait. Hold pipette like this.
Follow her whatever she says. If it is supernated, you should take only supernated. Don't disturb the bottom. If you disturb, your yield will be disturbed.
Yes. Very slowly you have to take. Slow. You are not taking. You have to press the pipette, no? Then only it will come. Remove once. Wait, I'll show you how to use pipette. Hold pipette like this. Done. Now you see this again.
Whatever the solution, either supernated.
Now you can't take. The more you wait, the more it will start solubilizing. So you should not wait. You should immediately take it slowly and keep it. This you will press. Now I'll show how to use pipette. This is pressed already. See here. Now you will keep. Slowly you will release. See now it came. But I don't want to use because it will contaminate. If you don't know this pipette using, you can ask. This is the basic. If you don't use it properly, nothing will come properly. Now add 40 microliter of NaCl. NaCl is there. See, 5 molar NaCl is there. Now where is the protocol? So we did till here. Carefully cover this. We covered the upper aqueous phase, and then we have to add 40 microliter of 5 molar NaCl. So okay. This is 10 microliter. First you see this is 10, this is 1000 ml, this is 200. So you have to see this. You have to keep on rotating. You have to add NaCl 40 microliter. So I'm just telling them how to. This is 40. So there are different pipettes. One is 2 to 20 microliter, and one is 2 to 200. one is 1000 ml, one is 5 to 10. So now if you need, this is 20 microliter. If this is 20 microliter, you can directly take this or use this 200 one. This is what it is written. 100, right? Now you can use this. Set it to 40. Show me how will you set. No, first you press this and then rotate. This is locked. So why it is locked? So that when you're taking, it should not move. Press on this and then rotate. Continuously, along with that. First you have to press, then it will be unlocked. Then you can change. No? Okay, this is 40. Now I 40 microliter. And then 500 microliter of isopropanol. First adhesive, then isopropanol. You added okay. इन्होंने ऐड कैसे isopropanol था? इनको बताओ. This is 40, right? This is 1000. Oh, yeah. You have to hold it here. So you need 500. Now set it on 500.
It's already 1000, so we have to set.
Yeah, we have to set on 500. 500. It is going reducing. It is increasing. Yes. Now you set it on 500. You added 40 megaliters of NaCl. That now add isopropanol. How much it is written? 500. Then you add 500 microliter of isopropanol and incubate it for 10 minutes, I guess, at room temperature. Room temperature. This is awesome. Press only this. We have to incubate it at room temperature. Yes, yes. What it is written in the protocol? In the room temperature. See the timer also. Okay. After adding, what you guys doing? Added? No, timer. Then it's not timer. 10 minutes. Just a little what in between you can do. Like after 3 minutes, again once.
You can take 100 to 80 nanos. Fine, nothing I'm doing here. Keep it there. There is no details of you will take a it should be having 20 okay.
Everyone 5 minutes, 10 minutes here. 5 minutes. Hours 10 minutes. 10 minutes.
So any doubts in the meanwhile, during the time of incubation, you can ask regarding this experiment or in your lab, whatever the experiment is. Fine. You can ask me. We have incubation time, lot of incubation time. Right now, what is the step it is going on? 30 minutes, one more 30 minutes. It's done.
Incubation after adding NSCN and isotoping.
Oh, now phase separation will happen, right? So that step we should be very careful. You need to take only the supernatant. If you take the contamination part,
That's done.
that part is done.
NSCN
So have you carefully taken the supernatant, right? This is very crucial. That supernatant step is very crucial.
That's now you take it.
Yeah.
How much time you guys done?
a good question. Even when I was doing for the first time, the question is she did the cDNA synthesis in her lab. She got bands like multiple bands like this. She's worried. She's like feeling like many bands are there contamination. But I am saying this is a perfect cDNA. I'm saying this is a perfect cDNA should appear like this. This many bands should come. So how many of you are agreeing with me that this is perfect results for her? But if you agree, then you should say why. That's why I think no one will come. But this is really good. It will be like this only. For any cDNA, if you are doing, you will be getting multiple bands. But the reason why you guys going to say? No, it's nothing to do with purity. No, the question is, she has RNA. Like us, she also isolated the mRNA, and she went for cDNA synthesis. And she went one more step to check the cDNA bands are fine or not in the gel, 1% gel she ran. But she was very sad that she got multiple bands.
Maybe it's a half synthesized complementary DNA which has no molecular
No. Why a cDNA should ideally be multiple bands, not a single band? But yeah, if it is a single band, same molecular weight, these are different. You can see the molecular weights are very much different. Like lower range to higher range, it is there. Can see? No, this is actually ideal results, I say.
This is half run.
Yeah. Still, even though you run, they will separate it very good.
I did that also. Keep your tubes, I'll keep for 10 minutes. Actually, the picture is not clear.
Okay, no problem. No problem. Just for. Yeah, that's what. Let me answer. Let me try. Someone can able to answer this. Any answer from you? You understood my question, right? What about any answer? But have you ever thought in this perspective that is it? Have you run first of all the cDNA samples in gel? No. Only DNA, cDNA. It's only cDNA. If mRNA is there, then you will find a smear. No contamination. That's I'm sure. Because she would have confirmed in 260/280 ratio, as well as she would have run the gel before the cDNA. She would have confirmed the purity is fine, as well as she would have find the mRNA is intact. Then only she would have gone for the cDNA synthesis. So you can eliminate contamination. There is no contamination.
Different components which are present.
Different
Polymerase
components.
and all that.
No. you still mRNA. mRNA. It's a global mRNA. So you have 100 genes. 100 gene mRNA will be same size or different size. So if you are changing 100 gene mRNA to cDNA, should it be the same size or different size? That's what she got it. Those are the cDNA of different genes. So ideally, if you are converting to cDNA, you'll be having multiple cDNA, multiple bands. It depends upon your gene size. So you understood. When I ran first time, I also had the same doubt, and I was very much worried, and I was asking my seniors and asking my friends. Then I came to know that. So ideally, a cDNA should be a multiple band. Then only you have isolated proper RNA. If you are getting single band, that's a PCR, not a cDNA. Okay. I think now they are in the process of centrifuging. Okay, fine. Okay, let's break for a tea. We'll come back 4 o'clock. Fine. Okay, just have refresh yourself. We will start after that. Where is Yusuf? Ah.
Randy, Randy, where is this? Now, discard this malginated and add 1 ml.
Guys, just follow the protocol. So what is the step now?
Is it on?
It's on. Discard the supernatant, right? Or
Yes, yes. Discard the supernatant.
yeah, don't disturb the pellet. Take only the supernatant. Discard it.
75% gravity, we have to do that also.
Because now the steps are very sensitive.
Discard the supernatant. Now we are examining.
So let's go in the group wise. First group 1 will add everything. So I'll be bringing this entire thing to the group 2, then group 3. So in that way we'll do so that the crowd will be managed. So there are components: water, forward FP stands for forward primer, reverse primer. This is the master mix. So show me the protocol.
5 microliter.
So pipettes and tips. So you need
20 microliter.
10 microliter. 10 microliter is more ideal.
10 microliter.
10 is not that. 20 is okay. 20 is fine. You can use 20. So master mix is 5.
5
Up to 5
So you set it to 5 microliter and add to PCR tubes are here, right?
2 to 20.
Take it in the
80.
PCR stand. Do you have a PCR stand?
No.
Just Shreya,
12.
can you give a PCR stand? That RT-PCR boxes, no? Oh, here, we got it. Shreya, we got it. You can keep it in ice and label your group name. You keep 4 tube so that again and again no need to open.
Total 4 units.
Yeah. And close it and keep it in the stand. Let them label for them. So see this and start adding. So PCR master mix. This is we are making for 10 microliter. Okay. I will be making a small change. The forward primer is 10 picomolar concentration. The working I made.
Okay.
So I mentioned 0.6 microliter, but 0.2 is more than enough.
Okay.
So we will add 0.2 microliter. So and generally when you make, you always make multiple samples, 5 to 6. 0.2 is very less. For now we are adding 0.2, but in your later stage in experiment, you always 5 to 6 samples. Generally you make PCR for bulk samples, right? So make a master mix. 5 samples means multiply everything into 5. Accordingly you make, then aliquot 9 microliter and then add the template. So that will reduce your error, chance of error. Okay. So now we start with master mix 5 microliter. PCR tube she kept it there, I think. You label one tube, group number 1, right? Just add 5 microliter of master mix. You can find it mix.
Master
Yes,
mix.
Mix.
Yeah, that will be.
That is master mix. 5 microliter. Slowly now. Keep it in.
Next is
Next will be forward primer. You will add 0.2, 0.2.
This is 20.
yeah. Will this go to
This is 5.
point 2?
This is?
0.2 it is not.
There is 10 microliter pipette is there?
Yes.
Can you give it?
This is not.
Use the. This will be point.
0.52.
Okay.
Point 2.
The 2 microliter pipette is there, no? Point. No, they need 0.2. 0.2. Fine.
0.2 microliter.
0.2. Very less. So very gentle you should take. Very
Reagent
careful.
PCR right now, no? PCR.
I think this is on 2.
Be very careful. This is 2.
2.
This is 2, but you need 0.2. You need to set it to 0.2. Show me. Yeah. So for FP.
Forward. Every time you are adding your forward and reverse primers with a dot in the sides, or you directly add it to your
Dot in the
master
sides
mix?
also ideal. Finally you need a centrifuge. It's a remember. You need to remember. That's it. You can either write it in a book and tick it if you forget, or you can add it side to count the dots. But take right amount. If you didn't take enough amount, no, you will not get the amplification.
This is not right.
Otherwise, take 0.3 microliter. That's it. If you feel it's not ample, take some. Show me. Show me. Just hold this. No, they are not taking.
Yes. Maybe this tip.
Give me some.
Change the tip.
Not just
No.
taking. Maybe that pivot is.
Why? It was working.
Is ఎంత మైక్రోన్ ఇది పెట్టారు 10 మైక్రోన్
It's not taking.
better.
Okay, let's add
5
minimum what it takes.
0.5.
Yep, 0.4
How
at least it
much?
takes. 0.45. Okay.
So with the tip only we can mix.
Which one in this? Yeah. But while changing the primers, you need to change the tips. You add it directly inside. Okay. This is very low quantity. No? It's better to add it to inside.
Reverse prime.
Yeah, reverse primer now. Add. Make sure that little quantity is coming. That's very important.
Yeah.
Is that right?
Yeah, there is.
Yeah, fine. So that's enough. So now we added primers. Finally, we need to add DNA, but water you calculate. Use your phone.
Okay.
5 microliter, we added 5 plus 0.45, 0.45. How much water you need to add, you need to calculate accordingly. 5 plus 0.45,
Actually pipettes are
another
not
0.45, plus 1 is for DNA.
Plus 1.
DNA, minus 10. No.
So this will be
This will be the
3.1.
total reaction. Remaining how much?
3.1.
3.1 microliter will be water. Add water of 3.1.
set.
3.
No, that is 2. Maximum it can go till 2 only.
So take 20.
3.1 you need it, right?
So 20 microliter.
Yeah, 10 microliter to 20 microliter.
20,
10 is
20.
there. 10 is there. Use this.
10?
10 microliter. You need 3.1 microliter.
Okay.
It was locked, no?
Wait, is not.
Whatever?
This is not.
No, it is locked. No, you opposite side you went. You should have not gone. That is 10 microliter. Like, be careful. Pipettes are very costly. You want how much? 3.1, no?
3.1.
Okay, 10.
RNS free water.
Yeah, give me.
There.
No, here it's
Here.
Water is there.
Oh, also
And
there.
this
That's
same.
also aliquoted, same thing. I just aliquoted here.
Okay. Just give a vortex, done.
Yeah,
right?
we'll finally vortex everything. Yours is done, no?
Done.
Yes.
Just pass this pipette, 2 pipettes, you just take this. You labeled right your group name. That's very important.
Let
Take
it
this.
pipette.
Pipette I'm taking.
There.
Pass it to the group 2. Which is group 2?
This is no. Yours.
This is two.
Okay. Let's go with group 2. Yeah, just open the protocol manual. So now we are going to make the master mix for PCR. So they mentioned what is the master mix? Primers, forward primer, reverse primer, template. Template is we kept there. We need to take that. And finally water. Here I am going to change one thing. Instead of 0.6, we are going to add 0.45 microliter. Means we can it will get amplified at very low concentration. So I reduced it. If you add, yeah, if you are taking forward 0.4, you should take the reverse also. We cannot change the ratio. It will give a problem. Okay, 1:1 ratio you should take. So you are also in group 1. Okay, fine.
And the enzymes and other things that would be.
Sorry, enzymes I mentioned. There will be major 4 components for any PCR. One will be the master mix. Master mix will have DNTP, polymerase, those things with a buffer. Apart from that, you need to add the primers, forward primer, reverse primer. and finally will be your template. And you need to make up the volume with water. If they are lesser than the 10 microliter, you need to make up with the water. So now we are going to add 5 microliter of the master mix. I kept the master mix. This is mix, no? This is master mix, green color one. This has the polymerase, and FP stands for the forward primer, and this is reverse primer, and water to bake up. DNA we will add finally to everything. Okay. So who is gonna add now in your group? Decide. Maybe one. Each one can add one, right? So let's start with, what's your name?
Sharmila.
Sharmila will start with polymerase. So you need to set it to 5 microliter. Can someone? So this is 10 microliter. You can see the volume. Any pipette, you take any pipette, in the cap they would have mentioned the lower to the higher volume. So 0.5 is the lowest. This pipette can take minimum of 0.5 microliter and maximum of 10 microliter. Okay, 0.5 to 10 microliter it can take. So now our requirement is 5, right? So 5 we are taking. So this will be 10. If you are rotating and it goes to 10, that will be maximum. We are in somewhat in between, 5 microliter, and this is the lock, so that mistakenly the volume should not change, no? So they mentioned this is lock. If you want to change, you should pull it down, open the lock, then you need to adjust. Okay, now we are locking. 5 microliter I have set. So Sharmila, take the tip. You need 10 microliter tip, not that one. There will be small tips. What's his name? Go.
In these 5 components are there, no? Can I add one? This one?
Yeah. No. That is not compatible for 10 microliter tips. That is for 200 microliter. You can use the small tips. And while taking also, as I said, first is single press. Take it, then remove, then oil drop. Yeah. Now mix. Yours mix. And where are the tubes? PCR tubes. PCR tubes. So you took 4, no? Give it to her. I don't have gloves. Yeah. Keep it inside. Keep it here. And label your group 2, right? Keep one more for the other group. No. Wait.
Less
Let her label
primers, so
first.
we are doing only one tube.
Which one?
Less primers primers are very expensive, so we are only doing
Yeah,
one.
master mix. Unnecessarily, why to waste resources? You just label it here with group 2, right? No. It's for online. It's connected to online.
Okay, you are telling me to catch it, so I am putting it together.
Okay. Anyway, it will be recorded there. Fine. So master mix, you take Sharmila, right? You take 5 microliter from there. No, it should not
Take
touch
other
any
tips.
tips. Yeah, change the tips. Discard here. Yeah. Take a new tip. Open that. Yeah. It should not touch again. No. You keep it inside the ice first. Fine. You keep the tube inside first. Now you take the tip. Yeah. Now you open. Yeah. Now also be careful. Yeah. Yeah. I think food is, we are not providing sufficient food for you. I think.
Every day the double.
Yeah. Take 5. Yeah. Drop it. Drop it inside. Yeah. Fine. Fine. Anyway, we will spin it. No problem. Fine. You should not have air bubbles. Next time when you make, no, you should never have a air bubbles. Air bubbles will disturb the. They did it fine. Okay. You are doing multiple process, no? That's why air bubbles will get introduced. So okay, fine. Next time we'll see.
Another one.
Which one? Other sample. You can give it to.
One more tube. That bubbles are there, no?
No, they let the other group will do. Fine. You add the. Next is a forward primer, right? EFP. Yeah. there is no specific RPM. It's like it should be like a average speed, like 5000 RPM, and 1 minute is more than enough, sufficient for this. I'll see. It's like a vortex. It's a mini vortex. That's it. Now we can see the pellets are. You can see the calyx 100 at the bottom, right? Along with the debris. The supernatants will be separate. So you need to, for your DNA template, you should take the supernatant. Yeah, pellet should be discarded. That's it. So I will be adding for each one of you, because in order to save the time, I will add respective samples DNA. Can you bring the master mix? PCR master mix. Yeah, what we made for 3 groups, right? 3 groups, right? Yeah. Give me 1 ml pipette, 1 microliter pipette.
Sir, 1 ml?
1 microliter, the blue, yeah, no, another one is there, no? This one. This one. Yeah, use a gloves and take it. So now I'm going to add each group. So which is group 1? This is group 1, right? Group 2. This is group 3, right? This is mine, I don't only add. Okay. Here, this is group 1?
Yeah.
What is this?
Group 2.
Doesn't look like 2. Is it 2?
G2. G2.
G for?
Group.
Good. And this is 3. Okay, fine. Later on, you should not, if anything happened, then you should not blame us. Okay. Okay, I'm taking 1 microliter and I'm adding. Okay. Group 1 I am taking. I am taking only the supernatant. Okay. Oh, I forgot to set the pipette volume. Exactly 1 microliter. Okay. And I'm going to add to the group 1. Okay. Group 1 is done. Similarly, group discard box you can keep in here. What is this then? This is group 2, right? Okay. Instead of keeping here, I kept it there.
Write the name again.
Okay. Yeah. Marker is there. Can you give me a marker? Just write it group 2. See here, a lot of air bubbles. Air bubbles should not be there. It will hinder the amplification. So now, after I added the template now let's centrifuge for a short one. It's like a short spin.
Added 1 microliter template into each.
Each, yeah. For 10 microliter reaction, 1 microliter is more than enough. If you add more volume of sample, no, it will hinder the reaction. So you should check there is no bubbles, right? Right. Okay. Bring this sample there. Cycles you note down. But TM for my primer is 57 degrees Celsius. Okay. So maybe one after the other groups. Again, it will be clumsy. I'll show you everyone, but one after this. Yeah.
51 temperature.
57 for this primer. Okay, this is group 1, right? Okay, here there are many things can be done. As I said, this is denaturation, annealing, extension.
And initial, there will be, for any PCR, you will keep initial denaturation, which is a long time. 95 degrees Celsius, you will keep for at least 3 minutes to 5 minutes. That's the time where all the template will be separated. But after that also, you need to keep a cycle. This cycle will be repeated for 30 or how many hour cycle you are keeping. Yeah, you can take a, otherwise I will move out, later you can. Fine. So 95 degree for 30 seconds, followed by 67, but this is an earlier program. I will be modifying here according to me. Wait. So options I need to go. It's gradient. I don't need a gradient now, so I'm going to change it from gradient to the particular TM. I need 57. 30 seconds is more. 30 seconds. 30 seconds. Means each cycle. For each cycle, every cycle 30 seconds they will denature.
Okay.
And then 57, that is my annealing temperature, followed by my extension time. The for this polymerase 1 kb should be 1 minute. So my product is 1100 base pairs. So I am giving roughly around 45 seconds.
45.
Okay. And the final extension will be 2 minutes.
Okay. 4 is in.
I am keeping 27 cycles.
27.
Okay. This is the program. Then I will be keeping inside and I will switching on. That's it. Fine. You send the next group.
Once I will open.
Yeah. Just open this. Yeah. Slowly. Pull it back slowly. Fine. That's it.
It should always be 25 or can it be?
No, 35 we can go. Maximum 35.
Minimum?
Minimum 25.
Then you can put 21 as a point.
Fine. 27 is the average. Yeah. Whatever. Any problem?
No,
Or you are
no,
seeing
no.
your own face. You are shocked. Okay. Still it's recorded. Okay. Yeah. Okay, so this is a program. As I mentioned, there will be 3 steps. One will be denaturation, 95 degrees Celsius, and this will be annealing, and then will be extension. And there will be always one initial denaturation and one final extension. But this middle one will be for 30, whatever the cycle we are setting, they will be repeated. The arrow mark shows these 3 steps will be repeated for 27 cycles. You can keep it till 30 or 35 according to your requirements. If you increase the cycle, the amplicon will, the concentration you will get more. That's a concept. So as you changed everything, so I kept 95 degree 3 minutes 30 seconds. My annealing temperature 57, that is specific for my primer, and 30 seconds. Extension, this extension time depends upon the polymerase. The polymerase which we use suggests that 1 kb means 1000 bases. It requires 1 minute. That's what we are going to change. For 1000 kb, 1 minute. We are going, our amplicon is 1100. So we need to keep 1 minute 30 seconds something, right? So 1 minute 10 seconds, sorry. 1 minute 10 seconds or 15 seconds for safe side. Okay. Extension we set it. Then we need to keep it inside and start. That's it. It will show the time, how long will it take to set it. Fine. You can send your next. Any doubts you can ask.
27 is round number of cycles.
This 27, right? Yeah, number of cycles. I reduced the cycle so that we can complete it earlier.
Like what the difference between the first, that is the 95 degree for 3 minutes,
Yeah.
and then it's only continuing.
Yeah, because initial denaturation we always give for long time. So once the primers can able to bind, for example, that I will be telling you, initially we are going to amplify from the entire genome. Once the cycle starts, the PCR fragment will be made. Only 1000 bases will be made. Then it does not require that much time, 3 minutes for amplifying. Your fragments become only 1000, right? So 30 seconds is enough. So first amplification is always longer because of the genome we need to find, primer need to find and amplify.
So how do we know that is 100, it is split into 100 base pair at one time?
Trying to run the gel. Molecular, you need to say amplicon length, right? That while designing primer itself we will be knowing. Accordingly we need to do. Yeah. I'll be telling you during there, like explain in detail. Fine. You can send the other group. Yeah, last group. Anyway, RNA. What about RNA?
Last step is washing.